Good morning. I'll try it again. Good morning.
Good morning.
All right. I'm Jennifer Farmer, Executive Director of the Friedreich's Ataxia Research Alliance. It is my honor to welcome you on behalf of FARA, the Muscular Dystrophy Association, the National Ataxia Foundation, and CureFA, our co-hosts for this Friedreich's Ataxia patient-focused drug development meeting. Joining us today, we will have more than 25 representatives from the Food and Drug Administration, more than 25 pharma, biotech, academic research partners, and more than 140 patient families in attendance here, and many more watching online, as this meeting is being shared live via YouTube. There's no assigned seating, and this is on purpose. Please don't be shy. Please reach out and meet somebody new today. Our goals for today's meeting: to communicate the patient perspective of Friedreich's ataxia, the symptoms and how those symptoms impact activities of daily living and quality of life, to describe what individuals with FA currently do to manage their symptoms, and discuss what types of treatments would be meaningful, treating individual symptoms, or more globally, slowing and stopping this disease. Give individuals with FA center stage in helping us understand their battle and need for an urgent army of supporters. I would like to begin today with a brief overview and introduction to Friedreich's ataxia. FA is a rare, inherited, progressive disease affecting 1 in 50,000 individuals. The neurologic features of the disease, loss of balance and coordination, most often the presenting symptoms during childhood, are 100% penetrant. It's a progressive disease, leading to loss of ambulation 6 to 8 years after diagnosis, and loss of physical abilities needed to do activities of daily living. The effects of FA go beyond the nervous system. It's a multi-system disease, affecting other organs such as the heart, pancreas, and more global constitutional effects such as fatigue. There's phenotypic heterogeneity in age of onset, rate of progression, and extent of non-neurologic system involvement. There are no approved treatments for FA. Management and treatment recommendations include screening for scoliosis, cardiac disease, and diabetes. Supportive therapies, such as bracing and physical therapy, are used to manage some of the neurologic symptoms. Individuals with FA are at risk for premature death. The average life expectancy is 35 years. The cause of early mortality in FA is most often related to advanced cardiac disease, congestive heart failure, and arrhythmias. FA is caused by mutations in a single gene, FXN, or frataxin. The common mutation is an expansion of a naturally occurring GAA repeat in intron 1 of the gene. Normal alleles have less than 30 repeats. FA alleles have anywhere from 100 to 1,500 repeats. FA is recessive, so both FXN genes have mutations. There is mutational homogeneity, with more than 96% of individuals being homozygous for GAA repeat expansions, and a small percentage of individuals having one GAA repeat expansion on one allele and a point mutation elsewhere in the gene on the other allele. The GAA repeat expansion leads to epigenetic changes which result in transcriptional silencing of the gene and frataxin protein deficiency. A few important points. Individuals with FA are not frataxin null. They are frataxin deficient, making less than 20% of normal frataxin. Carriers have about 50% of normal frataxin and are asymptomatic. There is no aberrant or abnormal protein. The protein that's made is normal and functional as the mutations in the intron. The gene is silenced, so not enough frataxin is produced. The size of the GAA repeat is correlated with age of onset and measures of disease severity. The longer the GA repeat, the earlier symptom onset, the more rapidly progressive, and the higher risk for cardiac involvement. The frataxin protein is an essential protein, highly conserved evolutionarily. And while it's a nuclear-encoded gene, its primary function is in the mitochondria. Frataxin is involved in iron-sulfur cluster assembly. So frataxin deficiency leads to iron-sulfur cluster deficiency, which then has a cascade effect on various mitochondrial processes, such as altered metabolism and decreases in energy or ATP production. There's also thought to be excess iron in the mitochondria, which contributes to oxidative damage. When we think about treatment or therapeutic strategies, we have to cast a wide net. We know that we will need more than one drug or therapy to treat and cure FA. Downstream of frataxin deficiency, compounds that reduce oxidative stress, improve mitochondrial function, boost ATP production, or improve cellular metabolism are actively being evaluated in clinical trials. Of note, many individuals with FA take vitamins or supplements that are thought to reduce oxidative stress or improve mitochondrial health. While there's high unmet need in FA, we continue to evaluate therapies that might improve symptoms, such as anti-inflammatory agents or drugs known to impact neurologic function, such as pain, spasticity, or motor control. Researchers have been looking for ways to enhance, stabilize, or replace frataxin, attack the disease at its root cause. A lot of work's been done to evaluate strategies to increase transcription, small molecules such as HDAC inhibitors or RNA oligonucleotide approaches. AAV-mediated gene replacement therapy holds significant promise. In the past 5 years, there's been great progress in demonstrating feasibility in animal models, such that several academic and industry programs are actively doing the critical and important pre-IND and IND-enabling studies to bring programs to clinical trials. As research advocacy organizations, we fund a lot of early stage and basic research to drive drug discovery. But we also look at the entire drug development process and identify ways where we can be effective at expediting and improving the likelihood of successful outcomes. Building clinical research infrastructure, Friedreich's Ataxia Global Patient Registry, we established in 2005, has more than 3,000 individuals enrolled. In 2004, we supported and participated in the first evaluations of disease-specific neurologic rating scales and performance measures, and established a clinical research network and natural history study that's enrolled more than 900 individuals with annual evaluations. Promoting advocacy and patient engagement in the pre-competitive space with activities and programs like the one today are our highest priority. We believe that when we have all stakeholders, patients, researchers, industry, government partners, sharing experiences, insights, asking questions, engaging, seeking opportunity, we will make the most progress. In summary, Friedreich's ataxia is a disease that can be clearly diagnosed with genetic testing. However, early signs and symptoms that present during childhood are often dismissed until the progressive nature of the disease demands attention. The devastating outcomes and lack of treatment demand urgency. On behalf of FARA, MDA, NAF, and CureFA Foundation, thank you for being here today so that we can have all key stakeholders assembled to learn more intimately about the patient experience and apply these insights to development and advancement of treatments for FA. Our next speaker does not need a whole lot of introduction. He is well known to nearly all in the room and online. My dear colleague, Ron Bartek, co-founder and president of FARA.
Is she absolutely and awesomely spectacular or what? Good morning, everybody, and thank you so much for being with us here today. As most of you know, our little family is one of thousands grappling with this disease every day. And I'm going to tell you a little bit about our family experience, only because that experience, as you know, is illustrative of many of the experiences your own families are having. When my wife Rachel, who's right here, and I met on Capitol Hill here in Washington in 1990, she had these three little boys. On New Year's Day of 1994, dressed as cowboys, they walked their mother down the aisle. And when the question was asked, who gives this woman to this man in holy matrimony, the three bears, as we called them then, said, "We do." And there was not a dry eye in the house, not a dry eye in the chapel. We were a happy and healthy family of five, but our lives changed dramatically in 1997 when our son Keith, then 11, was diagnosed with FA. We had the other two boys tested. Byron was clear, got neither mutation. Keith got both mutations, obviously, and Stuart, our youngest boy, was a carrier like his parents. And we'll have to be conscious of that fact as he approaches the period when he might decide to start a family. Rachel and I sat at our computer almost immediately to learn about FA and tearfully embraced as we realized that our son was going to get very sick very fast and would not have the future that we had so dearly anticipated for him. We read about the FA symptoms that Jen was very good in telling you about, and we saw that they would have a life-shortening impact. We saw no treatment, no clinical trials, no organization devoted entirely to supporting the research in this rare disease, and decided that Rachel could look at that diagnosis, though, and look back and say, we saw signs early that we didn't recognize as signs of FA. For example, there was Keith's vision. Even as an infant, his eyes would quite often bounce back and forth. His balance, as you can see in this photo at age 5, he was already struggling against imbalance. His feet are much further apart than those of his brothers. His coordination. Early in elementary school, Keith was very much interested in karate and signed up for a class. He dropped it after only a few lessons because he just couldn't keep up with his classmates. We also noticed that he was experiencing awkwardness of gait, stumbling, falling, and difficulty with his handwriting. Rachel remembered thinking the eye jerks were simply a peculiarity that would disappear over time. We thought that the incoordination was a normal function of rapid growth, and we worked with him on his penmanship, thinking he really just needed better technique. And I'd been trained by the nuns, so I thought I could help him with that. There was also the psychological impact that you'll be hearing about some today from our patients. Keith had done very well in his first few years of school. He was very bright, very positive, very good-humored. As we began to notice the issues with his coordination and handwriting, however, we saw also that his grades were beginning to decline. One tearful moment, found his journal of that time and read an entry when he was commenting on his declining performance. He actually wrote to himself in his journal, "Maybe I'm just a dummy." That, of course, broke our hearts. Later, when he did get the diagnosis, we saw that he did regain some of his sense of humor, some of his cheerfulness. His grades started coming back. Basically, he regained his self-esteem to a large part and rebounded as he realized it wasn't his fault. He was not a dummy. The only good news Rachel and I saw that night at the computer was that the FA gene had been identified the year before. That, combined with our experience here in Washington in both the executive and legislative branches and the fact that we lived only 45 minutes from the FDA and the NIH, armed us with the conviction that if we were able to, that we should be able to do something about this disorder that we just learned to spell. So we concluded that if we established an organization devoted entirely to supporting the research in FA, could raise sufficient resources and awareness, and collaborated with everyone interested, our organization could make a real difference. We sought out 3 leading FA scientists, 5 other parents like ourselves, 1 adult patient, and formed FARA and got it incorporated in 1998. 6 months later, we used an NIH workshop grant to conduct the world's first international scientific conference on FA. 80 investigators from around the world gathered at the NIH for 3 days, joined by 1 other patient advocacy group, the National Ataxia Foundation. At the same time, no industry partners came. They all said the same thing: you're too rare, you're on your own. And there were no clinical trials underway at that point. I asked one of the investigators who was leaving the meeting that Sunday afternoon why he and so many other scientists were spending 3 days with us at the NIH with such a rare disease. "Easy, Ron," he said to me, "we think we can get this one." That chokes me up every time I say it. I've been saying it for 15 years. So. We assembled the field for the 5th time near London in 2015. At that point, 350 scientists participated, along with 12 industry partners and 10 other patient advocacy groups, including 2 of our partners here today, the MDA and NAF. We also had 9 clinical research programs at various stages of development at that point. And here we are now, less than 4 months away from our 6th International Scientific Conference on FA, surrounded by hundreds of patient families here and online, along with some of our academic investigators, industry colleagues, collaborative advocacy organizations, the MDA, NAF, and the CureFA Foundation, and government partners, all working together with a strong sense of urgency and making real progress together. Of course, as we were making all this considerable progress, Keith's disease was progressing as well. And we saw that he was acquiring all of the symptoms that we'd read about. At age 11, when diagnosed, he already had considerable hypertrophic cardiomyopathy, thickening of the walls of the left ventricle, as well as scoliosis and these square wave jerks in his eyes, causing issues with reading. At age 13, he participated in studies that indicated FA patients have higher iron levels in key nerve cells and suffer significant mitochondrial dysfunction. He also added his blood donation to the 50 samples that Rachel was collecting that brought the Coriell Cell Repository's collection from 5 to 50 FA cell lines. Very important in the research that's been conducted since. He underwent at age 15, he underwent 5 hours of spinal fusion surgery for scoliosis, implanting metal rods along his spine. By age 16, Keith progressed to full-time wheelchair use, was experiencing cardiac arrhythmias, and taking prescription heart meds. He also participated in the FARA study at NIH with 14 FA patients across 18 months that was really responsible for developing the clinical test that launched FARA's natural history study, in which hundreds of you, of our patients, in your annual exams at our clinical sites around the world, have built the database that's been so essential in establishing the clinical outcome measures for our clinical trials, and that have established without a doubt that they can reliably and quantifiably report on the activities of daily living, your ability to perform activities of daily living, and the impact on your quality of life. At age 18, Keith was diagnosed with type 1.5 diabetes, requiring insulin injections, and he was experiencing serious hearing problems. By age 21, Keith lost much of his speech clarity and volume, all of his peripheral vision, and some of his central vision. His loss of core stability led to the need for waist and chest straps on his wheelchair. He experienced increased difficulty with activities of daily living and required a personal care assistant. By age 23, Keith was almost entirely dependent on others and was losing his ability to communicate. His speech was very difficult to understand. He'd lost his ability to manipulate his keyboard, so he asked us to type his emails and Facebook messages to his friends. By September 2009, his hypertrophic cardiomyopathy had migrated to become dilated cardiomyopathy, and he was admitted to the hospital with congestive heart failure. In January 2010, after 5 months in and out of the hospital, Keith's congestive heart failure took his young life a month after his 24th birthday. With the challenges he had faced, Keith had not been able to complete college, though he was very bright. He got straight A's in his senior year in high school and in his 1 year of college. He had not experienced falling in love and having children, though he would have been a wonderful husband and father. Keith was gracious as he faced death in his final days. He looked at us and said, "I know you hope to be in time for me. I know you're going to keep going till you get it. You'll be in time for some of my friends." Keith lives on in our hearts and is still doing his part in research. He'd always wanted to travel more than he was able, and now, 7 years after his death, he's been to Europe and Australia, and as part of the very active FARA biorepository, his tissues have played important roles in our research, and his cells are being used in high-throughput screening assays. So you can see our family is one of thousands of contemporary families whose lives have changed with this diagnosis. And we've all come to realize that our lives are not the only ones changing. We see the lives of our brilliant academic investigators, industry colleagues, and government partners like those with us here today from the FDA changing as well. We see they've all come to appreciate and encourage the increased expression of the voice of our patients, and that they genuinely care about us and want very badly to help us succeed. We still don't have an approved treatment, but have developed a robust treatment pipeline that Jen showed you, full of promising therapeutics advancing through clinical development and profoundly beneficial therapy programs nearing the clinic. And FA patients, you as FA patients and advocates have played a major role in all of that. Once isolated, we now are all in, all together, all the time. Once hopeless, we are now completely confident, as you are. The scientists who once told us, we think we can get this one, now say to us, we know we will get this one. With all of us working hard and urgently together, as we always do, we know we will treat, cure, eliminate, and ring the closing bell on Friedreich's ataxia. Thank you. Now I'd like to introduce our opening FDA speaker, Dr. Wilson Bryan. Dr. Bryan, MD, is board certified in neurology and neuromuscular medicine and has seen a good number of our FA patients. He came to the FDA in 2000 and now serves as director of the Office of Tissues and Advanced Therapies in the FDA Center for Biologics Evaluation and Research, otherwise known as CBER. Dr. Bryan and his office colleagues, who will be responsible for reviewing all of our community's proposals in gene and cell therapy, also hosted FARA's excellent meeting on gene therapy with our gene therapy companies and universities only 4 months ago. It was our very first CBER meeting, and he was our host and chair. We greatly appreciate his willingness to provide opening remarks for this meeting today. Dr. Bryan.
Good morning. I'm here representing, as Ron mentioned, the Office of Tissues and Advanced Therapies and the Center for Biologics and the Food and Drug Administration. I want to talk about education. My own medical education began about 40 years ago, when I was accepted to medical school, and for me, it was a great honor and privilege to have the opportunity to become a physician, to learn medicine. I want to say a few words about one of my patients. About 20 years ago, I was a neuromuscular specialist at a medical school in Texas, and I was a co-director of an MDA clinic. And of course, my responsibilities included seeing many patients with neuromuscular disorders, including Friedreich's ataxia. But I also had the responsibility of teaching medical students, interns, residents, neuromuscular fellows. The only way to learn medicine is to talk to patients and to examine patients. So every now and then at the medical school, I would ask for volunteers from among my patients to come to the medical school to help me teach. And there was a patient, I'll call him Mike, that's not his real name, he always volunteered. He was about 40 years old, with Friedreich's ataxia, in a wheelchair, and had a beautiful smile. And he almost never came to clinic. He almost never came to clinic. I'd go for years without seeing him in the MDA clinic. And he knew that at that time, medicine just didn't have a whole lot to offer him. But he had a whole lot to offer medicine. And over the years, he helped to teach hundreds of medical students and future doctors, as well as interns and residents. At the FDA, my colleagues and I, we recognize that we have to continue to learn from patients. And Congress knows this too. So back in 2012, Congress passed a law called the Food and Drug Administration Safety and Innovations Act, or FDASIA. And there's a section of that act that calls for increased patient input into the regulatory process. Since 2012, the FDA has held approximately 20 of these patient-focused drug development meetings. But we don't have the resources to have meetings for every disease in every group. So we really appreciate that the Muscular Dystrophy Association and FARA and the Ataxia Foundation and CureFA have gotten together and taken the initiative to sponsor this meeting. Because in a meeting like this, we'll learn things that help us to think about the design of clinical trials, to think about what outcome measures and endpoints are meaningful to patients, and to help us to balance benefits and risks when we're thinking about approving a new product. I think every doctor has some patients that are special, that stay in our memories, like I remember Mike. The patients are always with us. We may have had great teachers, brilliant teachers in medical school, but we know that most of our education comes from the patients. Patients at the FDA, my colleagues and I learn from patients, the patients who participate in clinical trials, the patients who serve in advisory committees, and from the patients and caregivers who come to meetings like this. So again, I'd like to thank the organizers of this meeting for putting this together, a meeting that will give us the opportunity the privilege of continuing our education by listening to you. So I thank you.
Let me ask if Dr. Moshinsky's in the room. Hearing no response, I'll turn it back over to Jen. The absolutely no.
So I would like to introduce James Valentine, who will be our moderator today. James Valentine is an associate at Hyman Phelps and McNamara in Washington, D.C., where he assists medical product industry and patient organization clients in a wide range of regulatory matters, including new drug and biologic development and approval issues. Before joining the firm in 2014, Mr. Valentine worked in FDA's Office of Health and Constituent Affairs, previously the Office of Special Health Issues, where he facilitated patient input in risk-benefit decision-making and served as a liaison to stakeholders on a wide range of regulatory policy issues, including helping launch the Patient-Focused Drug Development Program. So I don't think we could find anybody better to help moderate us through our day. James, thank you.
Thank you so much, Jen. Hyman Phelps and McNamara is very proud to represent FARA, and I'm very honored to be here with you today to be your moderator. As we begin our portion of the agenda today, where we're going to now be turning the mic over to patients and caregivers to hear about your experiences with FA and your perspectives on and preferences on treating FA, I wanted to give a little bit of an overview of why it is we're here today and how we're going to go about providing your input to FDA, as Dr. Bryan just so eloquently described. As Dr. Bryan mentioned, FDA about 5 years ago developed a more systematic way of gathering patient perspectives on their condition and on available treatment options. The patient perspective was recognized to help inform FDA's understanding of the context for assessing both the benefits and risks of potential new drugs. And it is critically important to inform FDA's oversight both during the development of drugs as well in its review of marketing applications for new drugs. So I believe the total of these meetings that FDA held on its own will reach 24 by the end of the series of meetings that they're hosting this year, which started back in 2013. And each of those meetings, 24 meetings, has been focused on a specific disease or disease area. After each of these meetings, FDA has published a Voice of the Patient report, which is a summary of the meeting that relies heavily on the actual words that were spoken at the meeting, including many of the quotes of the patients and caregivers that participated in those meetings. And these reports are very important as they provide information that's useful to FDA staff, but also beyond FDA. It's useful to researchers and drug developers to help understand the patient voice beyond just those particular meetings. In December of 2015, FDA announced the opportunity for patient groups to host these externally led patient-focused drug development meetings. And today is one of the first of those meetings, and we have quite a few FDA officials present, despite this not being on their own campus, so that we thank them for making the journey from White Oak here to be with us today. And we thank all of those that are also listening on the live webcast for their participation. Like FDA's meetings, the meeting organizers today, including FARA, will generate a Voice of the Patient report, which it will submit to FDA for FDA's consideration. So today's meeting, we've organized it into two topics. which we will in turn address. The first topic is how FA affects your daily life. We're going to be asking you questions about what symptoms related to FDA are most bothersome to you. We're going to ask how these symptoms affect your ability to do specific activities and how they might have changed over time. And we're also going to ask you a little bit about what worries you the most about living with FA. In our second topic, we're going to then switch to talking about your approaches to treating FA. And this is beyond just drug treatments that you might use, but also lifestyle modifications, diet, exercise, medical procedures, pretty much anything out there in the universe that you might use just to make your day a little bit easier living with FA. So we're going to ask you what you're doing to manage burdens and symptoms, how well those treatments work in addressing those symptoms, and what you consider to be a meaningful improvement from treatment. For each of these two topics, we're going to use a variety of different ways to discuss with you these questions and elicit your input for FDA to hear. The first way is we're going to hear from a panel of patients and caregivers that are living with FA. The purpose of the panel is to set a good foundation for our discussion and also try to hear a range of experiences with FA. After the panel, we'll then open it up to get everybody's input, both in here in the room and online, by having you answer a series of polling questions. The purpose of these questions is to aid in our discussion. Whether you're in person or online, you'll be able to use your cell phones, tablets, or laptop or PC computers to respond. I'll explain those directions on how to actually use those once we start our first set of questions in a couple of minutes. We do ask that only patients and their direct caregivers respond to those questions. We'll ask those that are here from the healthcare community, industry, FDA, and the like to please not respond to those, so that way we limit our responses to those that are patients and caregivers living with FA. The third and final part of each of our topic discussions will be a broad dialogue with all of you patients and caregivers that are in the audience today. The idea here is to build on the experiences that were shared by the panel and discussed in the polling questions, as well as we'll ask you to actually raise your hand to respond, and we'll bring you a mic and ask that you state your name before answering. We only have a limited amount of time today, so I'm sure there's going to be things that you're going to want to say that we might not have time to get to, and that's okay. FARA will be collecting additional comments on the topic questions in written format by email for 30 days after the meeting, and at the end of the meeting, we'll remind you how you can do that. Since we are relying on all of you to provide input to FDA, I wanted to establish some ground rules just to make sure that we're treating today in a fashion that will be conducive to FDA being able to get the most input that it can from our discussions. We encourage all patients and caregivers, including family members, to contribute to the dialogue. Our FDA colleagues, who are sprinkled throughout the audience. Actually, could I have our FDA colleagues here raise your hands and just maybe wave to those around you? Okay, I see a whole bunch scattered pretty much all across the back of the room. So our FDA is here to listen. We won't be calling upon them to answer questions or provide any input. And we also, our researchers, clinicians, and industry colleagues are also here to listen. We know that your views you're going to speak to today are your own personal opinions, and so we ask that you please respect each other as we have this dialogue, whether or not your experiences or preferences are the same or different from others that are speaking today. So that's what we'll be working through over the course of the next couple of hours. We're now going to shift into our first set of polling questions, which are to collect some demographic information from you, from those that are participating both in the room and online. So we have instructions here, and I'll walk you through them. So pull out your whichever device you plan to use, whether that be a cell phone, tablet, or computer. One kind of piece of housekeeping before we get started is, obviously you're going to be using your phones for polling, but we do ask, out of respect for each other, that you do silence it. And since you're going to be using your phones throughout the day, if you do need a charger, just raise your hand at any point, and one of the FARA staff will come by and bring you a charging block. Okay, so for how to participate, we have 2 ways to participate, and it's the same for whether you're online or in person. However, if you are online, or maybe if you're a patient or caregiver sitting towards the back of the room, might have a little bit more difficulty seeing the screen and the questions once we get to them, I'll recommend that you use the second method, which is the URL rather than the text messaging. So first I'll explain how to use the text messaging way for responding. It's pretty simple. All you need to do is text FARA PFDD to the number 22333. Once you do that, it'll give you a response letting you know that you're logged into the system and able to respond to the questions. For single answer questions, you'll have the responses will be displayed on the screen with letters. All you need to do is enter the letter corresponding to the desired answer and then hit send. Some of our questions will require multiple answers. For example, choose up to 3, or choose your top 3, or choose all that apply. For those questions, we ask that you enter each letter separated by a space or a comma, and then hit send. It's important that you don't just text all of the letters with no spaces or no commas. That won't go through. If for some reason you submit an answer that you want to clear and be able to re-answer the question, just type the word undo, UNDO, and hit send, and it'll clear your answer, and you'll be able to respond again with your responses. If you prefer to use the URL, this option will actually display the responses on your device. Just go to the URL that's listed, www.pollev.com/FARA-PFDD. If you go to that link, you'll be able to just directly participate. Here, if you for any single answer questions, you'll just select the desired answer directly. And then for multiple answer questions, you'll be able to select up to the amount that you're allowed to directly on the screen. And at the very bottom, there's an option to clear your responses, which will wipe out all of your responses and allow you to re-enter your response. So as we move from one question to another, know that you'll only be able to respond during to the question when it's displayed, and we will be displaying the results as we move through the questions. So hopefully you've had a couple of minutes to follow these instructions and get logged in. If you have any questions or are having trouble, just raise your hand now, and someone from FARA will come by and help you either set up the texting or URL browser. So any hands? I need to send anyone right here up front. We have a gentleman in the peach shirt. It looks like orange, maybe peach. Display the first slide again. Could we display the first slide? Oh. With the instructions. Sorry, maybe it's the second in the order. Yeah, I think that's the slide you were looking for. Okay. So give everyone a second to get this worked out. The good thing is once we're all set up here, there'll be nothing new you need to do each time we go to a new set of polling questions. you'll just be able to pick up where we left off. All right, I know we still have a few people getting this set up, but some of the first questions we can leave up for a couple minutes and just to get started. So if we can move to the polling questions. So the first polling question is just, what is your name? So here you'll just enter your name and send it as a text message, or if you're on the browser, you can just type it right in. Once I get the signal that we're all good to go, we'll move to the next question. So thank you for your patience. Wish I did. Are we good? Okay. Did he hit submit already? Do you know? Or is he still typing? We're good. All right. So we'll move into our next question. So here we're just asking if you can provide your email address. Go ahead and type that in and hit enter. Looks like we had about 145 people answer the first question. So between here in person and online, we have 145 of you participating, which is a fantastic number. looks like the results are slowing down a little bit. I'll give you a couple more seconds to get in your email address. So now we're going to move into some of the questions that require you to look at responses and provide letter responses, our actual demographic questions. So the first question is, where do you currently reside? A, northeastern U.S., B, southeastern U.S., C, midwestern U.S., D, southwestern U.S., including Texas, E, California, F, northwest U.S., not including California, G, Canada, H, Mexico, or I, outside of North America. So not surprising, the majority of our participants are from the northeastern U.S., so geographically close to us here. Our next largest contingency is in the southeastern U.S., followed by Midwestern. Looks like we actually have some people participating from Canada and outside of North America as well. So our next question is, do you live in A, city, B, a rural area, or C, a suburban area? All right, so more than half of our participants are from suburban areas, about a quarter are from rural areas, and about 20% are from cities. Our next question is, which of the following best describes you? A, I have FA, or B, I am the parent or caregiver for someone with FA. Okay, so it's pretty close to 50-50. Slightly more of our participants are parents and caregivers. Slightly less than half are individuals that have FA. For our next, some of our next questions, we're going to be asking questions about the individual diagnosed with FA. So if you're a caregiver that's completing this, please answer on behalf of the person with FA and not on behalf of yourself, and you'll see what I mean. So the question is, at what age were you diagnosed with FA? So here we're interested in what was the age of the person that you're responding for, whether you're the individual with FA or a caregiver. The answers or responses are A, 0 through 5 years of age, B, 6 through 10 years, C, 11 through 15 years, D, 16 through 20 years, E, 21 through 30 years, or F, if you were over 30 years of age when diagnosed. looks like about 60% of the individuals being represented today were diagnosed 15 years and younger, and then in the 16 to 20 year range, just under a quarter of you, and then the remaining being 21 years or older. The next question is, how long ago were you diagnosed with FA? So were you diagnosed less than a year ago for A, B, 1 to 2 years ago, C, 3 to 5 years ago, D, 6 to 10 years ago, E, 11 to 20 years ago, F, more than 21 years ago, or G if you're unsure? All right, so as the last results trickle in, I think we can see that about a third of our participants today were diagnosed 11 to 20 years ago, and then a little under 20% both for the 6 to 10 years ago and more than 21 years ago. We do have some individuals that were diagnosed shorter than that, 12% 3 to 5 years, 12% 1 to 2 years ago, and 8% of you less than 1 year ago. And none of you were unsure about how long ago. So here, this again is for the age of the individual affected with FA. So if you're a caregiver, respond for the patient and not yourself. How old are you? A, 0 to 10 years old. B, 11 to 20 years old. C, 21 to 30 years old. D, 31 to 40, E, 41 to 50, or F for older than 50 years old. Question. Sorry. It's a good question. So I'm going to defer that question to so the question was, if the individual with FA has passed, what age should you put down? And the answer to that is to, just do not respond to this question. We can skip this question. Okay, so it looks like we're getting to the final responses here. It looks like about a quarter to a third of you are each either 11 to 20 years old, 21 to 30 years old, or 31 to 40. At 10% or under are those that are 0 to 10, or 41 to 50 years old, or greater than 50 years old. So that concludes our demographic questions. Before we move into the two topic discussions that I described to you earlier, we have a video to share with you. So we had the opportunity to spend some time with two of our FA families. As you can imagine, not all those that are affected are able to participate in this kind of meeting in person. And so we wanted to share with you their stories. The Delorios from Rhode Island and the Helms from Montana. They generously allowed us to capture a day in their lives on film. In the film, they openly show us what life is like with FA on the daily tasks that able-bodied people might take for granted, as well as the impact on progression over time. Discussing one's challenges, one's fears, is never easy, but both families share their realities with strength, grace, and hope for the future.
A typical day for Matt is he'll start saying, I want to get up, time to get up. So we get him. We use the lift that's in the ceiling to get him out of his bed. So we raise the back of the bed up and we can get him onto the lift easier. First step is to go into the bathroom. We wash him in the bathroom if he's not taking a shower that day. Get him partially dressed. It's a lot easier to put his pants and shoes and socks on while he's on the toilet. The next step is to go into the wheelchair. That's when we'll get his top on, deodorant, finish bathing him there. Then from there, it's off to the kitchen for breakfast. Pills.
It's a complicated process because we have to take his blood, give him his pills with yogurt. He has a compromised swallow. He's had 3 swallow studies. The recommendation was to use thicket for all of his drinks and all of his food. So he does take his pills with yogurt, and that's a little difficult because some of the capsules and the vitamins are large. They're not easy to take with the yogurt. And he has his breakfast. He can eat most of his meals by himself. He loves to watch sports, golf, college basketball, hockey, baseball, depending on the season. He keeps in touch with his friends. He has a lot of good supportive friends. He's very interested in music. After college, he did DJing a lot on his own, which was great.
Matt would love to participate in trials, but he has his ICD and he has the diabetes. And with that, it would not give them a true reading on what they need to find when they do a trial. And his age has a lot to do with some of the trials that he can't participate in. With our race, he feels like he's contributing with raising money to help other people get into the trials and hopefully someday find a cure.
I do everything to make sure that the younger kids that have a fight don't have to go through what I did in him myself.
What about when you get older?
Well, I'm going to have my own castle and I'm going to be rich.
7 years have passed since the filming of the Ataxia, and Dylan is now not able to get out of his chair and walk anymore. He has had scoliosis surgery, and he also has developed diabetes. He wears an insulin pump and just recently automatic blood glucose monitor. He's more dependent on us and for pretty much everything. He has trouble eating independently now. His dexterity makes that pretty hard for him, but he still has his personality and his huge heart. That hasn't changed.
Sienna is 10 years old now. She'll be 11 next month, and she really shows no symptoms at all in her face. She's extremely active. She loves softball, she ice skates, she rides a bike, she does pretty much anything she can to be moving all of the time, and we encourage that because the more she can do now, the longer she's going to be able to do it.
My first memory was when Dylan could still walk, we'd always hang out. And then when he got into a wheelchair, he just was really mad. Sometimes I'll think like, what's gonna happen when I'm in a wheelchair? I'm gonna have to quit all my hobbies. I'm gonna have to quit all my sports. That worries me.
If you can get a treatment in place that stops or slows the progression of FA and just let them be where they're at and let science catch up in other areas to start helping undo the damage and repair the damage, I would be happy with that. I want a kid to be happy, thankful for when the day is a good day and hopeful that tomorrow will be better.
So I'd like to quickly first invite up our first panel. But I want to take a second to thank both the Dolorios and Helms families for sharing their stories with us. And Matt, if you're out there watching somewhere, I wanted to, on behalf of all of us here in Maryland, wish you a very happy birthday today. So happy birthday, Matt. Okay. So now we're going to transition into our first discussion of the day, which is on living with FA. As our panelists get set up, I'll provide you some of the questions that are going to guide all of the portions of our topic 1 discussion. So the first question that we're going to be asking the panel, and then subsequently all of you to respond to, is, of the symptoms that you experience because of your condition, which 1 to 3 symptoms have the most significant impact on your life? Second question is, how do your symptoms and their negative impacts affect your daily life? Third question is, are there specific activities that are important to you, but that you cannot do at all or as fully as you would like to because of your condition? And lastly, what worries you most about your condition and the progression of your condition? And these questions are all printed on your agendas, and we'll display them when we get to the audience discussion. But without further ado, I would like to introduce to you our topic 1 panel. We have Frankie, Jack, Kristen, RJ, and Nelda, and they're going to each share with you some of their responses and their stories as it relates to the topic 1 questions. So, Frankie, take it away.
Hello and good morning. My name is Frankie Perzola. I'm 24 years old, born and raised in beautiful Los Angeles, California, with a family the size of most elementary schools and an inconceivable love for the game of basketball. I've always had a passion for any sport, really, especially now with an admiration and jealousy of all types of movement. At the age of 22, 1 year after I graduated from Arizona State University, go Devils, and my career set in sales for professional sports, with dreams as high as being the first female GM at the NBA, I was diagnosed with Friedreich's Ataxia. I was asked to talk about certain symptoms that affect me most, and to be honest, all of them shake me to my now unstable core. Remember years ago the magic 8 balls we asked questions to? You shook it and waited for the answer on that little triangle to pop up on the window. That is exactly how I feel about us FA patients. All of us are just sitting and uncontrollably shaking the 8 ball to see what symptoms and how severe they will be in our progression. Slurred speech, diabetes, scoliosis, loss of coordination in everything from your fingertips to your toes, or eventually becoming fully dependent on someone else for help on living your life, just to name a few. We don't know, and that is absolutely terrifying. After years of questioning my body's declining capability of physically keeping up with my peers in softball, struggling to stay on junior varsity in high school, the inability to walk a few blocks after being able to cross ASU's main campus for 4 years, not being able to climb a set of stairs without white-knuckling the railing, and the draining fatigue I would experience after simple tasks such as vacuuming, doing a load of laundry, or mopping the kitchen, I finally had the answer. The diagnosis, Friedreich's ataxia. There's two ends of the spectrum you can come from if you're an FA patient. Either you've had it since you were younger, most likely suffering the more severe symptoms. They've known FA their whole life. Or the place I come from, which is having a later onset of symptoms, living a normal life, and slowly watching my body deteriorate and struggle to do things I once did with ease. It took me 2 whole minutes to pick up a bobby pin off my bathroom floor the other day because I'm slowly losing control over my hips and arms. I would and will not let the bobby pin win, ever. Whatever way you look at it, both ends of the spectrum are completely devastating. No one can teach you how to deal with an incurable disease or how to handle the sound of your parents heartbreaking as they watch your body get worse every day. My father, who is 65 and retired, has to install handrails around my house to help minimize my falls. Watching that happen, knowing the rails are for me and not him, is extremely hard to process. Handicap placards and a countdown on time I have to drive with my feet are now everyday thoughts. Accepting that I have physically more in common with my 80-year-old grandmother and her friends than I do my own 24-year-old friends is honestly mind-blowing. Having her push me in a wheelchair, knowing it should be the other way around, has been hard to experience as well. Choosing between my cane, walker, and wheelchair, depending on the event or activity, and noticing more wheelchair days than before, is hard to accept and mentally debilitating. We need to address the progression and somehow slow it. Honestly, I haven't been this scared in my entire life, and it's the only time I haven't had a solution for one of my problems. I can't begin to explain the fear of watching her body physically declining, with birthdays now being markers of tracking my progression and how much worse I've gotten. It's time we break that magic 8-ball open and get some definite answers. We need to have some say in what happens to our bodies, because after all, we didn't have a say with having FA. Thank you.
Hello, my name is Jack DeWitt. I am 17 and I live in Howell, Michigan, outside of Detroit. My parents first noticed that I had balance problems when I was 5 years old. By the time I was 9, I had a walker and I was finally diagnosed with FA at 10. When I was 12, I had a electric scooter and I couldn't walk at all. When I was 14 and had my spinal fusion, I try to remain optimistic, but I know my symptoms from FA will only continue to get worse. My biggest problem with FA is fatigue, simply because it affects everything, every day. Fatigue really affects my school attendance, and I often miss a half or even a full day of school. On a day when I'm feeling really, really good, I can get through what most kids would think is a pretty boring day. I can manage my shortened class schedule and then either do therapy or homework at night. Never both. On my worst days, I don't even make it to school and just try and catch up with teachers through email. Just this year, I've missed 184 class periods. I try, but caffeine, it only goes so far. Now, I am 17, and my grandfather grew up in Kentucky, so it's no surprise that I became interested in rifles. Now, as you become more interested in these rifles, you get to the point where you start building them. This is not very easy when FA affects your fine motor skills. I don't know how many of you have opened up a rifle trigger group before, but there's a lot of tiny springs and screws that go everywhere when your fingers don't do as they're told. It is both incredibly demoralizing and very frustrating to see a hobby that I love being taken away from me. I see my fine motor skills getting worse every day when I play games like Call of Duty, while my friends are getting better and killing me over and over and over. My KD stats are getting worse. Also because of FA, I've had to give up on driving. During driver's training, I tried to drive with hand controls, but couldn't really see well enough or react quickly enough. For example, I couldn't see street signs because of the nystagmus that I get from FA, and I would be halfway through an intersection before I realized I needed to turn. However, I still have posters of a Corvette and a Mustang in my room because I am a teenager from the Motor City. Now, I mentioned that I'm 17, and that's the age when you start getting calls that make your parents worry. About a month ago, I got my first call from a Marine recruiter. I had to turn him down on account. However, I have always wanted to serve in the armed forces, but I guess I have other battles to fight. So I'm most worried about losing my vision because of FA. I feel if I lose my vision, I'll lose my remaining link to the world. It will take even more of my independence away, and I'll lose more hobbies and activities that I enjoy, like hunting, video games, and travel. I guess I miss seeing my friends' goofy looks and, alright, yeah, even my brother's stupid face. Thank you for giving me the chance to tell you how FA affects me.
Hello, and thank you for the opportunity. My name is Kristen Morrow, and my husband Ben and I are the proud parents of our kind and delightful 11-year-old daughter Anna, who was diagnosed with FA at age 9. As a 5th grader, Anna walks the half-mile to school each day with her siblings and neighbors. But this simple act best represents the symptoms and challenges that she faces living with FA. Anna's heart rate increases many mornings during this walk. We know that we just need to stop, take a break, and make sure that we provide ourselves with extra time. But these heart rate inconsistencies, in addition to her staggering gait and lack of balance and repeated falls, have led to her riding her recumbent bike to and from school starting in April. When asked by our family and friends how Anna is doing, we often reply that she's doing great, but we know that this is not entirely truthful. While she is in the early stages of progression, we have noticed a decline in the past year. The progression is especially pronounced when Anna gets fatigued. Her balance becomes extremely unsteady, she falls more often, experiences an increased heart rate, and migraine headaches. Upon returning from a trip across the country to participate in a clinical drug trial, she became so fatigued and so unsteady, she held onto my arm for nearly a week and then requesting a walker to keep at home. We recently traded bedrooms with Anna and her sister so she could be downstairs and not have to navigate the one flight of steps, as she's fallen numerous times. Anna's lack of balance is particularly more pronounced in the mornings, as well as more frequent episodes of an increased heart rate, which occur several times a week, sometimes with no forewarning or correlation to her activity. This has frightened her to the point of tears, which has also led to anxiety, especially when participating in sports. She doubts her ability to swim laps for swim team, to keep up with her lacrosse teammates during basic drills, due to her inconsistent heartbeat and lack of balance. We often have to coax her to participate, many times without success. She sits out, upset with herself for not participating in something she loves to do, but just cannot bring herself to do so despite her strong desire to participate. She's becoming more and more self-conscious of these difficulties, as well as more strongly objecting to wearing her brace for scoliosis, which is prescribed 22 hours a day. While the hope is that the brace will minimize her scoliosis progression, her curvature has increased by 9% in the last year and a half, and wearing the brace makes it very difficult for her to remain active. We've discussed the importance of the brace in hopes that by doing so it will eliminate the need for an extensive spinal fusion surgery, which leads to a painful and multiple month-long recovery. Anna, however, is dedicated to staying strong. It takes up much of her free time, but she dedicates herself to personal strength training sessions and aqua therapy several times each week, as well as daily rides on her trike. Anna is not fully aware of what lies ahead. She knows that FA is causing her scoliosis, her difficulty with balance, and can affect her heart. She knows that she may have to use a wheelchair one day, but that's really the extent of it. We live in a constant state of urgency, trying to fit in as much as we can into one day as possible, but while trying to retain its normalcy. How much longer will she be able to walk to school? Will this be her last season of lacrosse? Will she need spinal fusion surgery? Will she be able to live independently in college? How long does she have? These are questions we hope to shield her from as long as possible. Anna has a heart that's brimming with kindness. However, our greatest worry is that with FA and the mild thickening in her heart will worsen. We feel that there are ways to modify her lifestyle and accommodate her changing needs, but the heart condition, which are much more serious, as they can end her life, not just make it extremely challenging. The struggles she faces are almost unbearable to imagine. We know that by the time our daughter is my age, she could develop vision and hearing loss, lose her ability to verbally communicate, and even feed herself. But the most unbearable would be that she's not with us at all, due to the life-threatening heart condition that often plagues those living with this vicious disease. Anna is the strongest yet most tender soul her dad and I have the privilege of knowing. We will stop at nothing to ensure that she's able to live her life to the fullest, and we thank all of those who are doing the same with those living with FA.
Hi, my name is R.G. Mercure. The most common word I hear is huh, thanks to my dysphagia, and I barely hear that due to my hearing loss. Thank you for this opportunity to give a voice to managing this lovely disease, Friedreich's Ataxia. I'm not one to be so negative. I love to smile, but today must be an exception. I'm 31 years old and was diagnosed almost 20 years ago at the age of 12. Before high school, my only symptom that was noticeable was a stubble here and there, something only close family would catch. I was also diagnosed with a life-shortening cardiomyopathy, but I never thought twice about it, as image was a huge concern as a teenager. Progression in high school changed my walking and talking like I had a little bit too much tequila. Just imagine a giraffe in high heels on a windy day. The best way I can describe to you my early years of FA would be if you were drunk all the time. Now, don't get me wrong, you don't feel drunk. Your physical reactions are ataxic. The definition of ataxia is the loss of full control of bodily movements. If that's not what you'd call drunk, what's your definition? That realization comes from a time in life I felt so good about not having to ride the bus with all the cruel comments from other kids anymore. And one morning in 11th grade, sitting in my class, feeling accomplished that I made it into school fine that morning, and I'm called into the principal's office because a parent had called in reporting that I was drunk. My handwriting, eyesight, and cinema were all weakening. After graduating high school, I was forced to adapt. I had to use my wheelchair full time. If you asked me 10 years ago what symptom I have the hardest time coming to terms with, my answer would consistently change, just like this disease. I'm sick of always adapting as symptoms worsen. It is so exhausting. My heart is on the losing side of this battle. A premature death that you hear in all the literature is becoming apparent. I'm not scared of it, but I'm sure my family worries every day. But of all the symptoms that I experienced, one that had the most significant impact on my life would be my speech. Our most defining characteristic given to us is our voice. And to have that taken away from anyone is awful. I'm often talked down to or belittled because of how I speak. Too many times to count. I've tried to explain to people why I'm in a wheelchair, and they are just staring at me like they have no idea what just came out of my mouth. As you likely notice, my poor muscle control causes slurring and breathlessness. It took 10 years just accepting myself as wheelchair bound. Now I'm not hearing well when I once could, seeing well when I once could, and now then losing the ability to be understood. And that's a recipe for feeling worthless. I'm not asking how to adapt to this one. of painting a picture of patients just like me, losing the ability to communicate, losing individuality, and losing independence. And all I've ever wanted was to be involved, be accepted, and contribute to society. FA makes that almost impossible. Being rare adds a lot of extra work and added loneliness. The clock is ticking, and I'm fighting against so much. We have to get this one. Thank you.
Thank you for this extremely important opportunity. Before I start, I would like to say to those in the patient audience that I do not wish to cause you pain with the details I'm about to share. This is a rare occasion for me to explain explicitly what happened in the last few years of our daughter's life. Please remember that no two cases of FA are the same. 1 year ago, we lost our 33-year-old daughter, Rebecca, to FA. Our oldest daughter, Robbie, who also has FA, is here today, and our youngest daughter, Katie, is a carrier and married with a 1-year-old child. Becca was a precocious little girl who excelled at almost everything. FA symptoms started at about age 7, and by age 14, she was using a wheelchair. This was before genetic testing was available, so it was a waiting game to see what she and her sister had. Despite worsening physical limitations, Becca was a hardworking student and made her mark in academics. Much to our concern, she set her sights on the University of Notre Dame. In 2001, she graduated as valedictorian of her high school class in Texas and enrolled in Notre Dame without being able to take a single step. In 2005, she graduated on time with 2 degrees after having the time of her life for 4 years. The faith-based, service-oriented environment at Notre Dame had helped her achieve her goals. Unfortunately, her health had been failing over this same time, and by the time she graduated, the toll of FA was very apparent. She could barely type, had begun having difficulty with the enunciation of words and the ability to control her breath when speaking. She began choking and strangling and had to have thickener in her liquids. She required 12 hours of sleep and never had much energy. She became unable to bathe and dress herself, brush her teeth or hair, put on makeup, and take care of her toileting needs. Rather quickly, she also lost the ability to feed herself and hold a cup. She soon was not able to roll over in bed or adjust her position at night, and she couldn't scratch when she itched. The last 5 years of Becca's life were very sad and very difficult for her and us. She developed heart failure and took more and more medication to control the symptoms of shortness of breath, fatigue, low energy, and fluid retention. These physical losses were accompanied by social isolation and depression. We had moved while she was in college, and it was extremely difficult to make new friends. The academic environment, which had given her life its drive for so many years, had come to a screeching halt, and her alternatives were few. She could not make plans because she never knew how she would feel. But when she did feel okay, she would watch funny television shows that entertain us with her quick wit. She also would take her service dog to a library program for reluctant readers called Read to Rover, and sit on the deck with her herb garden or visit the farm of some close friends. It was being close to nature that gave her the most comfort. 3 years before her death, Becca developed a blood clot in her heart. Her health declined even more rapidly, and she started taking powerful blood thinners. We had to buy a ceiling lift and rent a larger wheelchair because of the amount of fluid that she retained. She developed gastric paresis, which caused great discomfort. We constantly tried to determine what she could eat and what medication she needed to handle the bloating and distension. Becca could no longer think clearly and was unable to focus. The depression worsened, and then the most painful symptom raised its ugly head. She began experiencing attacks of intractable spasticity, which struck with no warning, causing excruciating pain. She would have to go to bed with these attacks because her body became so rigid that we could not keep her in her wheelchair. Medication helped somewhat, but at therapeutic doses caused extreme drowsiness, slowed breathing, loss of lucidity, and psychotic episodes. Becca often worried about the other families impacted by FA and helped us with our fundraising efforts. She knew that we were fortunate that I had physical therapy training and that we were able to provide her with good medical care, but she also knew she was dying. She often expressed how sad it was for her that we were having such a difficult time caring for her. She knew we were hurting too and not being able to protect her. We tried as hard as we could to keep her as comfortable as possible. I will always wonder whether our care kept her alive too long and prolonged her suffering. By the beginning of 2016, she was ready to go, and by the time we called in hospice, she lasted 1 week, taking her last breath on May 21st, Friedreich's Ataxia Awareness Day. For her funeral service, which perfectly captured her sweet spirit, we had to go back 4 years to find a photograph that looked like her. Becca's last good day was a Monday in mid-May, just 5 days before she died. Kyle Bryant and Felicia DeRosa were in town for the screening of Farah's wonderful documentary, The Ataxian. Becca was able to be up for about an hour, and although she couldn't really see, she knew they were in our home and was able to visit with them. She seemed serenely happy that day. In closing, Becca would want you to understand the importance of learning everything we possibly can to help alleviate the suffering in any way for those with Friedreich's ataxia. My hope is that her story will help to inspire research that could one day lessen the impact of this tragic disease.
Wow. Thank you all for being so brave and being the first to share your experiences with us today. We're now going to transition into the broader audience participation on topic 1 about your experiences with living with FA. And we're going to do that by first going to the polling questions. So if we can have up the polling questions. I'll just remind those of you that are participating that if you're a caregiver to, again, answer on behalf of the individual affected by FA. So our first polling question is, please select the letter that best describes your stage of disability. And I'll read the responses. A, minimal disability, you're able to run or jump. B, symptoms are present but mild, you're able to walk and capable of leading an independent life. C, symptoms are overt and significant, but require regular periodic holding onto the wall or another person for stability or walking. D, walking requires a walker or other aid such as a service dog, and you can perform several activities of daily living. E, you are unable to walk, confined to a wheelchair, and can perform some activities of daily living that do not require standing or walking. And F, you have severe disability, you have dependency on others for assistance with all activities of daily living. Give you a couple more seconds to get in your response to this polling question. Okay, so it looks like about a little over a third of you, both in the room and online, are not able to walk, but you can perform some activities of daily living. About a fifth of our respondents are walking, but it requires a walker or equivalent. And then the majority after that are able to walk without such aids, but maybe need stabilization or less, and 10% have severe disability. Okay, so this is the first of a series of questions about your symptoms, and we'll have the same responses but we're going to ask you slightly different questions for each. So for this question, this first question, we want you to select all that apply of the following FA-related health concerns that you or the person you care for have currently. And the responses are A, balance or the ability to stand, B, issues with walking and regular falls, C, coordination in hands and arms and manual dexterity, for example, difficulty grasping, gripping, or holding objects, or fine motor skills. D is, the responses are a little bit off. So C is fatigue, D is diabetes, F is scoliosis or curvature of the spine, I'll just go off that. Sorry, F is cardiac condition, G is choking and or swallowing issues, H is vision loss, I is hearing loss, J is dysarthria or slurred speech, K is urinary issues and or bowel issues, J is spasticity or cramping or stiffening in the legs, M is pain, and N is other. So please select all that apply. You can put them all into a single text message. If you happen to want to add on others, you can send those in additional text messages. Give you a few more seconds to get in your responses to this question. All right, so it looks like we have quite a spread of the various FA-related health concerns experienced. Those that are most frequently experienced are those that we have up front here: balance and walking, coordination in hands and arms or manual dexterity, and fatigue. After that, our next most frequent are scoliosis, cardiac condition, dysarthria and slurred speech, and spasticity or cramping and stiffness in the legs. And then we have individuals with all of the other symptoms, with only a few with diabetes, and we also have a couple others, and we'll explore what those might be when we get to the moderated discussion. So if you mark down another, please remember that, and please raise your hand when we get to the audience discussion so we can hear about what that is. So our next question is the same set of symptoms, FA-related symptoms, but here we'd like you to choose up to 3 that most impact your daily quality of life. Responses are still coming in. Okay, it looks like the clear top 3 burdens that are most impact our respondents' daily quality of life are balance, walking, and regular falls, coordination in hands, arms, and dexterity, and fatigue. Although all of the other symptoms were selected as being a top 3 symptom that impacted daily quality of life, and no one said an other. Okay. So our next question is, which activities of daily life are most important to you that you are not able to do because of FA? And again, we're asking you to select up to 3. And so the responses here are A, moving independently and safely, or walking and standing. B, manipulating small objects. C, writing and typing. D, sitting unaided. E, driving. F, feeding oneself, such as cutting food and also handling utensils. G, personal hygiene, such as bathing or dressing independently. H, transferring independently. I, communication. J, understanding conversation. K, reading books, so, seeing a computer screen or a phone. And L, other. And again, select up to the 3 that are most important to you that you are not able to do. So the top activity of daily living that's most important to our respondents that they're not able to do because of FA is moving independently and safely, or walking and standing. After that, the next highest rated activity of daily life is manipulating small objects, followed by writing and typing. No individual said sitting unaided, and it looks like also no individual said reading books or seeing a computer screen or phone, but otherwise we had each of the other symptoms or activities of daily life reported, and we also have a few others, which again I'd love to hear about during the audience discussion. Our final question for topic 1 is, as a result, or as the disease progresses, development or progression of which of the following symptoms worries you the most? And again, select up to your top 3. A, being balance, walking, or regular falls. B, coordination in hand and arm dexterity. C, fatigue. D, diabetes. E, scoliosis. F, cardiac condition. G, choking and or swallowing issues. H, vision loss. I, hearing loss. J, dysarthria or slurred speech. K, urinary issues and or bowel issues. L, spasticity, such as cramping or stiffness in legs. M, pain, or N, other. So again, please pick up to your top 3 of those symptoms that as the disease progresses, which worries you the most. So it looks like the top symptom that worries you the most is cardiac condition, which we heard a little bit about in our panel. After that, it's the walking and balance and falling. From there, it looks like the next cluster that's ranked is coordination with hands, arms, and dexterity, fatigue, and dysarthria. And then it looks like after that we have every symptom being ranked as most worrisome to you, and nobody selected other. So then we have a question as a result of living with FA, which of the following are most significant to you? So please select your top 3. We have A, depression and or anxiety. B, social isolation. C, loss of a job or inability to get a job. D, modified work or school hours. E, trouble building or maintaining relationships. F, frustration. G, lack of hope for the future. H, loss of independence. I, financial difficulties. J, communication issues. K, loss of hobbies or activities. and L, other. So please select your top 3 that are the most significant to you as a result of living with FA. Give you a few more seconds to get in your top 3 responses for this question. It's looking like loss of independence is that symptom which is the result of living with FA that is most significant to you. Following that is depression and or anxiety and social isolation. Kind of the third tier would be loss of a job or inability to get a job, frustration, and loss of hobbies or activities. And all of the results of living with FA were ranked by some of you as being most significant and no others. Okay. So we're going to turn to our first opportunity to hear from those of you in the audience, our patients and caregivers that are here today. For those of you that are on the webcast, I'll remind you that there's the opportunity to submit written comments to these questions. And we will, even for those of you in the room, if you don't have an opportunity to provide your comments to these questions today, you can also follow up with written comments. We have FARA staff members around the room who will, if you raise your hand, bring a mic to you. Please wait for the mic to come to you so that those on the webcast are able to hear you. And then please state your name before you begin speaking. And please do so each time you speak, so that way when we go back and look at our recording and transcript of today, we're able to follow your comments throughout the course of the day as we try to write our meeting summary. So I'd like to start by going to our very first question, and I'd like to ask you which symptoms are those that really affect you now. In other words, which ones are the most burdensome to you and have the most impacts on your daily life. And specifically, I'd like, if you choose your top symptom or symptoms, to please describe why you chose that symptom and perhaps share with us an example of how that symptom has impacted your daily life. So don't be shy, just raise your hand and we'll bring a mic to you.
And speaking as the patient or the caregiver.
Yes, and again, this is open to all of our patients and caregivers and family members. And for the purposes of this, if you're a caregiver, you can also share your perspective of what might be most burdensome or significant to your loved one from your perspective as a caregiver, as well as burdens on you as the caregiver. We're interested in all of this. So, yes.
Good morning. My name is Paul Conans. My daughter is Brianne, and she's 39. Was diagnosed at 9. Her most significant symptom, because she's a very progressed FA'er, is vision. And she basically has told me that without vision, you have nothing. The isolation from the world is complete in her eyes. She also has hearing problems, etc. To just share with you, several weeks ago we were down at Stanford University Medical Center to get her baclofen pump refilled, and on the way back home in her van, she was sitting up front, and I'm driving along, and all of a sudden she says, "Wow, wow," and she does this occasionally, and I always go, "What?" You know, "What? Wow." Yeah. And she says, "It's wonderful." And I said, "Well, what's wonderful?" And she said, "I can see the landscape. And we're driving up Highway 280 between Palo Alto and San Francisco with the beautiful hills, which are still a bit green. And she has regained her her far distant vision. And she went for like 10 minutes looking out. And then her vision went away. And then she spent 20 minutes grieving, crying as we went up the road. So vision for her is her connection. Without vision, she can't see the monitor. Without vision, et cetera, et cetera, et cetera. Thank you.
Thank you, Paul. And can you just also, I wanted to ask, how does her vision change much from day to day? And can you tell us a little bit maybe about how her vision has declined over time, what that looks like?
Sure. Thinking for a moment. Her vision has declined reasonably linearly over the 30 years since she's been diagnosed. The last probably 10 years or so, her vision will kind of go in and out, and I ask her about that, and she says, well, it's like it's raining, or you're looking through a window with raindrops on it, so everything gets skewed because you're looking through the raindrops, and I have to kind of look around the raindrops, and sometimes those raindrops are moving across her vision, and that sort of thing.
Yeah. No, that's very helpful. Thank you very much for sharing. Yep.
Good morning. My name is William Stedman. I am 45, and I was diagnosed with FA when I was 17. I think my biggest problem right now is well, it's hard to identify one problem because they're all very intertwined. So I would say that what's most tangible is a problem transferring to bed and being able to lie down whenever I need to. And that has to do with being fatigued all the time, as well as trouble transferring, as well as requiring a PCA, as well as requiring a lot from my wife and my stepdaughter to be able to help me get to bed or get out of the chair when they need to. And well, that's the short of it. That there tends to be a lot of intertwining. All the areas are connected to each other.
Yeah. And so you're currently unable to transfer at all by yourself, or some of the time?
Yes, I can transfer with assistance. Assistance in as much as blocking my feet, so when I transfer, my feet don't slide out, and then lifting my legs into the bed, because I can do most of it with my upper body. But I slide down. That is a big problem.
Sure. And could you say how long ago, how many years ago it has been since you were able to do it unassisted?
Since I was able to do last was not too long ago. It depends a lot on the material around. Like, if I have a carpeted floor and I'm wearing shoes, then my feet don't slide as easily. And I can do it, and I used to be able to work out more in my upper body, and I was able to develop my triceps more, and that really helped me transfer a lot.
Sure.
But over the last couple of years, I've not been able to do that.
Right. Thank you so much, William.
Thank you.
Do we have up front?
Hi, Laura. I'm running into my
Of the great many symptoms I've experienced, the two main symptoms that hinder me the most are my poor manual dexterity and hearing loss. My manual dexterity is problematic for me not only because I am no longer able to feed myself, but because I have severe difficulty typing. Typing difficulty exclusively keeps me from earning my PhD, the one degree I still need to achieve my ideal job as a professor and a researcher. My manual dexterity problems make everything I do so exhausting and tedious, if not impossible, and are steadily getting worse. Being able to feed myself would greatly improve my quality of life as far as not having to rely on paid help and increase my personal freedom. I have done physical therapy for years until my ability to fully use my arms and hands eventually became impossible. Second in my perceived severity is hearing loss. Hearing loss to me means difficulty filtering out any noise going on in the background from a conversation on which I want to focus. In my University of Georgia classroom, I have severe difficulty hearing the students, especially those that sit in the back of the class. I have noticed that my hearing loss extends beyond the problem as with background noises because I unfortunately miss certain words spoken by people in my daily life. My middle sister's death was incredibly heartbreaking and challenging for me, personally. I could not help but think ahead to my own decline and at the end of my life and the choices I want to have made before my death. Living with Becca makes me realize how even sisters with the same disease are often affected by FA very differently. Becca's heart disease was more profound than mine is, but she experienced vision loss and severe spasticity, two symptoms I do not have.
Thank you, Robbie, and thank you. I see a hand blinded a little by the light, but I can see you.
Hello, my name's Jenna, and this is my husband, Spencer. Our 7-year-old son, Logan, has FA, and Spencer and I share the same sentiment as the majority of the responders who said that cardiomyopathy is probably, for us, one of the most stressful symptoms, the unknown of when is it going to progress. He was diagnosed with cardiomyopathy at age 4. But for Logan, I think the most serious symptom would be the loss of the ability to walk. First graders are fast, and you don't realize how fast until your son's left behind. Sorry. Okay, I got this. So we see him sort of getting to where the kids are, and then once he's there, they're running someplace else. Sorry again. Okay, I got this. And he just wants to fit in. So you can ask me a question. I think I can handle it. Yeah.
Thank you for sharing that. Do you notice, are there good days or bad days for his ability to keep up?
Yeah. There's been a couple of times this year where we've got a call from school that he fell asleep in the beanbag chair during story time because he was too tired and we picked him up. But he really fights every day because he just wants to keep up. And he has a twin brother who does not have FA, and that's a big motivator for him.
Sure. Yeah, thank you. And you do got this, so thank
you. Thank you. Sorry about the, I did it.
No, you did great. So I see a hand in the middle of the room.
Good morning. You want to do it? You want me to do it? Okay. Yes. Okay, now that we have collaborated, I will be speaking. This is on behalf of my good friend Jordan Howell. I do believe finding a treatment that would improve my hand dexterity would help me drastically. Being able to type words at a normal speed on my keyboard would improve my overall communication. Being able to move my manual wheelchair at a normal speed because my hands are able to grasp the rims would allow me to travel much easier. My hand improvement would allow me to eat 100% of my food without any assistance, and not having to worry about many issues would be huge. This would be a great example of how important it is to even just take care of one symptom. Thank you.
Thank you. Thank you both. All right, so I'd like to explore those of you that said that you experienced symptoms or burdens other than those that were presented on the slides, or maybe those that were presented but we haven't yet talked about either on the panel or in the audience. I know we have the mic, so you don't have to answer that question. You can say what you were going to say, but just to start to get you, the rest of the audience thinking, to highlight some things maybe we haven't talked about yet. But
my name is Scarlett. I'm 22. I did not have symptoms until I was almost 20, so I'm not very progressed yet, but the hardest symptom I have is just balance, where you can stand just fine and then out of nowhere you just fall on the floor. I'm in the gym quite a lot, doing a lot of preventative exercises, and that balance problem actually caused me to fall and fracture my L5. So now because of that, I have to live with the broken back. So it's just, I don't know, that's the hardest symptom that's affected me this far.
Sure. And Scarlett, are there any activities that you've already, even though you said you're not far progressed, but the balance issues have made you either stop doing or not do as fully as you used to do?
I don't walk unassisted. I do walk, but I'll take somebody's arm. But because of that balance problem, I can't really do that without assistance. So I do miss the independence.
Thank you. Okay. Did we have any takers on discussing symptoms or burdens that maybe were that other category, or maybe that we haven't really talked about? Oh, we have a hand in the back.
My name is Dawn Graysick. My husband and I have 6 children. They range in age from 5 to 12 years old. Our 12-year-old was diagnosed in December with FA, and we're awaiting laboratory confirmation for our 6-year-old son. And I think the depression and anxiety are probably the most significant for our family. My oldest does not remember ever feeling normal. Her symptoms started at age 4. She never was able to keep up with her peers or ride a bike. When she was in public school, her teachers were constantly asking her to do what the other kids did, and she would come home and tell me, I know I'm stupid, but I can't, which is why we began our homeschool journey. My 6-year-old is struggling very greatly now that he sees his sister, who was once independent, relying on her wheelchair. He's recognizing that he too stumbles and falls and is having a lot of the same difficulties. And he's very angry and scared and frustrated, recognizing what lies ahead of him. So I think the depression for her, the social isolation, and the anxiety for him, seeing what is coming and knowing that he can't do anything to stop it.
And is there, Dawn, is there anything from day to day that triggers the anxiety or depression more than others, or is it pretty consistent or constant?
He hurts all the time. We never knew why he hurt. We thought my dad's knock-kneed, so we blamed it on that. Oh, he's got dad's knock-knee, so he's sore. But I heard him playing with his brother the other day after a zoo trip, and his younger brother said, my legs are sore today. And I heard Josiah, the older, say, yeah, that happens sometimes, but when you're like me and you hurt every day, you just get used to it. And it really broke my heart to think that, that's been his life, is having sore legs. Right. And he notices he'll get frustrated and just scream because I can't button my buttons. Why can't I button this stupid shirt? Little
Sure.
things he used to do that he can no longer do.
Sure. Well, thank you very much. Come back to Paul and make sure I'm not excluding anyone up here. All right, Paul.
In the other category, and I think Nelda referred to it in her talk, And preface, you know, we're a diverse group here. A goodly number of you are in early progression stage, and Ron and I and many others here are confident that you all will not ever be in the late stage. My brand is in the late stage. Two in the other category, I don't believe that we're there. One is slowing of executive function, and the other one is hallucinations. And you can talk a lot about whys, whether it's my daughter's 30 years of increasing immediate focus on controlling each muscle of her body as she coordinates, or you could talk about the number of drugs that she has tried under supervision, or the supplements that she has taken and is taking. She takes more than 30 pills a day. So how do those pills interact and interact? Or FA apparently now, they believe, can get itself in the lower brain and have effects there. So in the cerebellum, what kinds of things go on? So those are the two other symptoms that would apply to the late progressed that I know of with my daughter.
Yeah. Thank you for adding on that. And we'll take the comment right behind you, Paul, and then we'll go to the back of the room.
My name is Marilyn Downing, and my daughter's had FA for 20 years. One thing that I really notice, and it's been difficult for us, is whenever she gets sick as a virus, it's always so much more involved than it is for her friends who have the same virus. And her friends will be over in 2 days, and after 2 weeks, she's finally starting to feel like herself, or she gets terrific leg pains as a result of a virus that she's had. And any type of invasive, any type of surgery, it's enormous for her. I don't know if the nerves just keep firing and don't stop. They don't realize that she's already recovered. Her nerves are still firing and causing her pain. We find that very difficult. So we always make sure she has a flu shot every year. But that's been very difficult for us.
Okay. And in recovering from viruses, what specifically makes it harder for her?
Extra fatigue, and pain.
Mm-hmm.
I'd say those two things.
Sure.
Difficult.
Okay. Thank you very much.
Yep.
I know we have a comment in the back.
That's Marilyn, right? Marilyn grabbed what I was going to say, or most of what I was going to say. You know, I think if you ask a lot of people, a lot of parents, you know, the walking, the heart especially, and the fatigue are really big. But to add on to what Marilyn said was that we've noticed with a lot of kids that have FA that if they get sick, it not only takes them a long time to feel better, but when Sean was walking, his walking suffered, and it seemed like he would have a decline, and it might take him a month or more than a month to feel like he was back up where he got. Other parents said sometimes their kids never quite got back up to where they were before. So anything small that affects, you know, like anything that we would shake off really fast just makes everything on that whole list worse. I know when the swine flu went around before they had the vaccine for it, we had several people on our parents group whose kids got it and were in ICU.
Sure.
So, I mean, it's just everything like that.
And when you said Sean's walking decline and then would go back up, could you describe what you meant by that?
Well, when Sean was walking, he was just, you know, walking like a drunken sailor. So we just noticed that he was more unsteady, more unbalanced, you know, just much more likely to fall, much more tired. I mean, all these symptoms get worse when they're tired, so having fatigue as a symptom is crappy because everything else, when they're tired, everything else is worse. You know, the talking, the walking, the fine motor coordination, the swallowing, everything. So, yeah, so, you know, just almost everything on that list would be worse for a while. And you would fear oh, good. You were in kind of a plateau, and now we're hitting the next step. And most times he'd bounce back up from it. But like I said, some parents, their kids never quite bounce back up to where they had been before the sickness.
Sure. Okay. I think we have time for one or two more comments. So we have one right here.
Good morning. I'm Sean Baumstark. I'm 36-ish and living with Friedreich's ataxia. I was diagnosed at the age of 25. One of the, I don't remember what question specifically I answered other two, but I was one of the others. Much like a few people in the room, I'm still fairly independent. I work full-time. I live alone. I'm able to drive and have a lot of my abilities. Still, one of the other things that scare me, and not because I live in a rough neighborhood or anything, but, I can't run. And that's okay. It's a stupid hobby anyway. But what if I were to run into confrontation? I can't protect myself very well. And I try to be smart with my work sometimes. But what if a dog were to chase me or chase after me? Worse than that, what if an individual who thought I was a drunken sailor thought I would be easy to take advantage of or steal from or whatever? That scares me. That's another category that, it really doesn't have anything to do necessarily with a symptom, so to speak. But we're not always going to be surrounded by people like in the room today. So how do we protect ourselves when we find ourselves in a corner or in a spot where we might be alone?
That's really important. Thank you. All right, we'll go to Sean.
Hi, my name is Sean Anderson. I was diagnosed when I was 16, and I would also like to add that I actually was attacked by our neighbor's dog once, and nothing I could do, because I couldn't walk. So now I carry a kid or a stick and I play baseball, I play golf, so that might come in handy someday. So my two biggest problems, one is the cardiomyopathy. I currently have dilated cardiomyopathy, and it makes it so that I have a very hard time staying active and for any long period. And especially around here when it gets hot and humid, I basically have to stay indoors now or in the air-conditioned car. I'm a very big fisherman. Anyone who knows me, that's all I talk about is, what I'm catching, that kind of thing. And that's outdoors. So, it's not only the heart for that, but it's also the accessibility, because I can't walk. I used to, obviously. But I can't anymore. And being in a wheelchair, I realized there are very few places now that I can fish. I've lost over 90% of my spots that I really like. So, let's see here, I made a list. So, with the manual wheelchair, I'm lucky enough to be able to drive still. Very lucky. And I have a wheelchair van, and I can just transfer myself in and out of that seat. But when I go to stores, which really there are very few, because I can't roll my manual wheelchair because the dexterity in my hands, and I get my fingers a lot when I do try, and I really don't want to break one. I have a scooter, which I call SIFA, S-I-F-A. I'll let you fill in the first part. The last is Friedreich's ataxia. The first part is a little bit rude. It's my sense of humor. But SIFA is much too big for stores. because it's kind of an off-road style, but not really, kind of moderate, because I do a lot of fishing. But of course, that means that there are very few stores I can actually go into. And I noticed that a lot of stores also, even with the regular wheelchair, they're built for people who can walk, and there's not really a lot of room in the aisles because they put things in the middle of them. So those are my two biggest problems: having a heart that sucks, and also not being able to walk anymore or be immobile.
Sure. Thank you. So I think we're going to have to go to our break now, but don't worry. We'll have another chance for a discussion, and we'll be talking about approaches to treatment in our next session, but obviously coupled with that is obviously what symptoms or burdens you're attempting to treat. So hopefully if you had something to say, you can bring it up in our next session. But for now, we're going to take a 10-minute break, and we're going to return at 11 a.m. For topic 2. So thank you all for your participation. If our panel 2 panelists could make their way to the stage. I know, you guys are so good. Yeah, we're ready. Keeping me. All right, if we could all get back to our seats, we're going to get started with our second panel. So our second topic for today is on current and future approaches to treating FA. And so we're going to be exploring a series of questions on approaches to treatment. And I'll let you know what those are before we then hand it over to our panel to start helping answer some of those questions. So if our colleagues in the back of the room don't mind taking a seat, we'll go ahead and do that. All right. So our questions for topic 2 on current and future approaches to treatment that we're going to be asking you all to answer, and our panel is going to kick off our conversation with, are what are you currently doing to manage your symptoms? How well do these treatments treat your symptoms? How well do these treatments improve your ability to do specific activities that are important to your daily life? And then kind of switching gears from talking about your current treatment, we're also going to ask you to then provide us your thoughts and your preferences about specific things that you would look for in an ideal treatment for your condition, short of a cure. So on that note, we have a panel that is going to be speaking to these issues. And we have Pat, Nick, Emily, Alex, and Mary. And I will go ahead and hand it over to Pat.
Thank you. Good morning. My name's Pat Ritchell, and I'm the father of 2 girls with Friedreich's ataxia. My daughter, Angelina, began having balance difficulties when she was age 9 and was eventually diagnosed in 2013 at the age of 11. Her younger sister, Samantha, was diagnosed a few months later at the age of 9. When we received our diagnosis, it was overwhelming because the first thing we heard is that there's no treatment, no approved treatment for FA. So we began to research and learn as much as we could about the disease, and we soon learned that, as you've heard this morning, beyond the gait and balance, there are lots of other aspects to this disease: cardiomyopathy, fatigue, scoliosis, diabetes, and later stages, vision and hearing loss as well. One of the first things we found out and learned was that managing FA takes a lot of effort and a lot of time. Both girls, after diagnosis, were also found to be presenting with scoliosis, and so we focused on trying to manage scoliosis through prevention. That's physical therapy, and that's meant twice a week, hour-long drives up to New Jersey, annual or sometimes more than once a year trips down to Washington for braces to be made or adjusted. And all in all, we spend roughly 400 hours a year just on prevention of scoliosis. We also began to look at what's available in terms of dietary supplements that might help. We've tried a lot. We've adjusted and tried to find things that work. We currently are taking idebenone, we're taking vitamin E, we're taking curcumin, and we're taking sulforaphane. All told, each girl takes more than 20 pills every day. But these are not tremendously effective. Progression continues to move forward. Angelina now uses a scooter to get around. In addition to supplements, and we look at exercise as an important aspect. The girls do physical therapy twice a week. They go to the gym 1 or 2 days a week. They do therapeutic horseback riding once a week. In the summer, they also swim. I think that exercise is a really important component of addressing the disease. While it doesn't halt progression, having the stronger muscles does impact your ability to transfer, to stand, and to compensate somewhat with balance and walking while you still can. The girls have been active participants in trials, both interventional and non-interventional. In the past years, the girls have participated in a total of 10 non-interventional trials to help researchers understand the disease. Unfortunately, there are not very many pediatric interventional trials, so only Samantha has been in an interventional trial. in the past 4 years. And I think given that FA is a disease that often strikes between the ages of 5 and 15, certainly I would very much like to see more trials in pediatric populations, or at least pediatric arms of trials, and have strong support from the FDA for interventional trials at that early stage. Due to the lack of trials and some preliminary results in an open label trial, the one treatment that's had some benefit has been interferon gamma that Angelina has taken. It did have a big impact on fatigue, whereas she would come home from school and nap nearly every day. Today she no longer naps and is able to complete all of her schoolwork. I recognize that we're at the early stages of the disease, and that while the girls have progressed, there remains significant deterioration in front of us. Angelina has lost her ability to walk independently, but she can still walk somewhat, and she's no longer independent in some activities of daily living, such as bathing and dressing. She has mild cardiomyopathy. Samantha is at an earlier stage. She's still independently mobile, and her biggest issue right now is fine motor skills. Eating, handwriting are both very slow and very messy. I think that the different stages of the disease present different desires in terms of what you're looking for expectations of a therapy. For Samantha, I'd like to maintain her ability to walk and improve her fine motor skills. For her to be able to handwrite an essay on an exam, to be able to take notes independently in class. Angelina has largely lost the ability to walk, and her cardiomyopathy is worse. I'm much more interested in a therapeutic for the cardiomyopathy that could stop progression or potentially even reverse it. I don't want to see her dying from heart failure in the coming years. For both of them, I think a medicine that reduces fatigue is very impactful to their lives and quality of life. In a perfect world, it would be wonderful to have a therapeutic that addresses all aspects of Friedreich's, or even reverse damage that's been done. But given the nature, multi-system nature of the disease, that might not be likely. And so I think that, from our perspective, there's a clear benefit to any therapeutic that can slow or stop progression or even reverse damage with any aspect of this disease, whether it be the ataxia, the fatigue, the scoliosis, the cardiomyopathy, and speech, hearing, and vision. I remain hopeful with all the efforts of the various folks here in the room and outside that we'll see therapies developed in the near future that can benefit my daughters. Thank you.
Good morning, members of the FDA, pharma and industry partners, researchers, parents, and friends. My name is Nick Carboni. I am 30 years old, and I am a patient living with Friedreich's ataxia. I am speaking with you today to give my perspective, how I manage symptoms, and what my hopes are for the future of FA research. Over the past several years, my routine as far as the management of my symptoms has not changed significantly. The over-the-counter vitamins that I take have been part of my regimen for nearly 20 years. Approximately 400 milligrams of CoQ10 and 1,000 IU of vitamin E daily. I also take 81 milligrams of aspirin per day. The prescription drugs that I take are Sotalol and diltiazem for my cardiomyopathy symptoms, such as increased heart rate, abnormal pacing, and high blood pressure. These treatments for my cardiomyopathy, as well as an ICD implant, treat symptoms which aren't even very noticeable to me, which is very frightening in and of itself. The ICD activates when my heart beats abnormally and rapidly. It rarely happens due to the medications, but I really have no warning when it does, and I don't feel any different as an abnormal pattern continues. Having the ICD has unfortunately disqualified me from several studies, despite my physical ability to complete the test. As far as physical activities to help manage my symptoms, I exercise at least 3 hours per week, aside from activities of daily life, which can prove to be a workout on their own. I always have been very active and have done what is possible to manage symptoms. I work one-on-one with a personal trainer at a CrossFit gym. We spend a lot of time working on functional fitness movements and adapting traditional CrossFit workouts. We also work on things that require my mental focus just as much as my physical stamina, such as walking, sitting up, and standing. This certainly helps with my activities of daily living, as I currently live on my own and I have to have enough energy and ability to accomplish things such as grocery shopping, cleaning, taking out the trash, cooking, and doing laundry. My work is also something that helps me manage my symptoms. I am a local attorney and politician. The pressure and reliance that clients and residents have on me makes me feel important, and also reminds me that I have to keep working on myself and doing what I can to manage and keep the progression of my disease to a minimum. All of these aforementioned quote-unquote treatments are how I proceed with my life, despite the lack of energy that I feel on most days. Without these self-directed treatments, I would have much more difficulty accomplishing some of the things that I'd like to accomplish. With the treatment or cure resulting from increased clinical research and trials, my goals and accomplishments might come sooner and would increase in magnitude. Some of these goals are having a family, buying and remodeling a house, pursuing politics on a national scale, and traveling. A cure is something all affected will look to. It is a difficult task to consider what would be my preference to treat short of that. To me, some of the most important things would be to walk freely and for a long distance again, to stand and address a jury, to walk up the steps of the U.S. Capitol, and to chase around the children that I hope to one day have. That's where you come in. More must be done for those affected by Friedreich ataxia. Thank you.
Hi, my name is Emily Young. I am 21 years old and a student at the University of Illinois. Members of the FDA, thank you for taking the time to meet with us today. In 2007, I met with a doctor who seemed to display little interest in what my mother and I had to say. I have no reflexes, we explained. That's normal. He shortly diagnosed me with scoliosis and said I needed to wear a back brace to contain the curve in my spine. In 2008, I was diagnosed with the FA. However, the scoliosis was one of the only FA symptoms I was having at the time, so I did not really understand how my body could just gradually become limited over time. The fatigue, the falling, the wheelchair, all of that sounded too far and unrealistic. I mean, besides having a back brace, I was totally fine. I was all too scared of the adversity I'd begun to see. Besides, None of the other 13-year-olds in my middle school class talked about losing the ability to walk, so why did I have to? Over time, it became apparent to us that I needed spinal fusion surgery to stabilize my spine, where the doctors were going to insert a titanium rod into my spine and screw it into place, making my spine virtually unable to curve. However, this benefit would come at the cost of most of my upper body mobility. In 2010, I had my first spinal surgery at Shriners. It was 7.5 hours. When I woke up, the pain was worse than anticipated. My nurses kept coming, asking if I'm ready for a walk, and I was thinking I'm not even sure if my legs are still attached to my body even. I mean, I was in so much pain. As the week progressed, it was painful in every aspect. I mean, your body hurts unimaginably. Your family is trying to live in a hospital. No one is sleeping. And the nurses are still coming in every day asking you if you're ready to take a walk. But eventually I went home and faced a near 3-month recovery. I was still in a little pain but manageable at the end. But I was ready to go into my freshman year of high school. No more back brace, just getting stronger. At my next checkup, though, my doctors revealed to me that a screw had come loose in my back, but they assured me a quick and painless surgery could repair it. Knowing that quick and painless surgeries actually do not exist, we let my doctors do the repair. The pain was less, but still extreme. The doctors added the rod up my neck, stabilizing the lower portion of my spine, but further immobilizing me. I was able to start high school on time. However, I felt slightly less mobile than my peers for the first time in my life. I felt restricted in what I could do. Looking back on the experience now, I can say that this was the first time I felt limited with FA. This was my first big symptom and my first step in managing FA. Little did I know, it was one of the more easy fixes than things I deal with now, as everything now does not have a clear and concrete solution. I spent the last 18 months trialing a drug I thought to be very beneficial, only to find out it is not beneficial enough to be an approved treatment. In the middle of my school semester, I had a full course load, a part-time job, and a social agenda that involves little sleep. But I was on a medicine that allowed me to make this my lifestyle. So now, 2 months without the medication, when I wake up feeling more tired and weak than the day before, and wonder, how am I going to make it through the day? These are my FA symptoms now, and I am still looking for a way to manage them. Thank you.
Good morning. Thank you for allowing me to share my experiences living with Friedreich's ataxia. My name is Alex Fielding, and I am 29 years old, living my life despite FA. At 26, I was given my genetically confirmed diagnosis of late onset FA after years of struggling up the stairs to my apartment and staggering through weekend hikes with friends. To my friends with FA, I'm lucky. I'm a late onset patient with a slower progression and the ability to walk. To healthy family and friends, I'm unlucky, battling an incurable disease that makes each waking day worse than the last. Some days I feel blessed, but more days I feel cursed. The only constant is keeping myself motivated to push through every day and live my life as normally as possible. With this attitude, I've earned my master's degree in chemical engineering, worked and lived independently across the country, purchased and now maintain my own house, and now serve as the VP of operations in my family's 55-year-old manufacturing company. While these milestones in my life are accomplishments to be proud of, they're also critical, and they become integral parts of how I manage FA on a day-to-day basis. Some of my best therapy occurs on the job. Work is a dangerous and exciting place for me. I'm constantly around moving machinery, working on tooling with my hands, and walking throughout the plant to manage all departments. For 8 to 10 hours per day, I'm walking, moving my body in unique ways, using my hands, and most importantly, not mentally focusing on FA. These daily activities help preserve my motor coordination and engage me mentally to keep from succumbing to depression. Unfortunately, all this activity is taking a toll on my body. My gait and posture have adapted to keep me walking, and I feel it in my ankles, knees, hips, and lower back. They are all in constant stress and pain. Last year, my neurologist identified minor scoliosis as a new symptom to watch. I now fight a mental battle to keep myself moving, but knowing that I'm destroying my body while doing so. For me, my motto has become, you'll lose it when you don't use it. And this is why I try to cook for myself when possible, since for me, I find dicing vegetables and grilling foods require a lot of fine motor coordination, and especially increasing effort to make sure I don't slice off my finger. I grew up playing music and continue playing piano, saxophone, and guitar to maintain finger dexterity. Unfortunately, this can be a frustrating chore as I continue getting worse due to my progression. In the short time since my diagnosis, I've participated in several research studies and one clinical drug trial. I found these experiences extremely positive. The studies were transparent with me, allowing me to evaluate my own risk associated with participating. But I found the greatest risk to be not participating. With no approved treatment or even lifestyle choices that are known to slow progression or improve prognosis, living with FA is a constant race against the clock, and participating in studies allows research to move forward and most importantly renews my hope for a future without FA. It's becoming harder to keep up with all my responsibilities at work and home and to keep doing the things that I want to do. Earlier this year, we revised our 2 to 5 year strategy for the business. It's already difficult planning ahead for a small business. It's nearly impossible when facing a degenerative disease that you know will continue pulling you back. Short of a cure, being able to objectively slow or stop progression would be invaluable to me. I want to plan my life with confidence, to be able to rely on my current abilities in the future without the fear of devastating complications such as diabetes, stroke, or cardiomyopathy. This, coupled with a sustained energy level, would allow me to continue doing everything I do now, to live independently, work a rewarding career, and contribute back to my community. I can adapt my life to current symptoms, stay active and engaged, but fear of what I know the future holds continues to cripple me, especially on an emotional level. Thank you for your time this morning.
Good morning. My name is Mary Caruso. I'm the mom of two amazing young adults who have lived more than half their lives fighting Friedreich's ataxia. As I began gathering my thoughts about what I wanted to say, I quickly realized that it was a struggle. I just couldn't find the words to truly explain what life is like. I realize now I couldn't come up with the right words because I never really allow myself to look at what life is like for our family living with this disease. As an optimist, I've chosen to deal with it and make the best of it. The reality, however, is that life is unbearable in many ways. The anger and fear that embraces us is constant. I could never sustain this battle if I allowed myself to think about it. So yes, life as we knew changed the day a doctor wrote the words Friedreich's ataxia, and nothing was ever the same again. The emotional drain on our family is something I never imagined. Sam, diagnosed at 8, is now 30. As Sam moved through school, there were struggles as friends became aware of the progressive stages of the disease. Between middle school and high school, Sam pretty much lost most friends. I kept his spirits up by promising college would be different, kids older and more accepting. He moved on to college, eager to begin a new chapter. One night, I got a call from him asking if I felt like having pizza. Sensing something was off, I immediately accepted the invitation, grabbed a pizza, and made the drive. As I walked into Sam's dorm room, I felt this immediate emptiness. As Sam explained, a planned trip failed to include an accessible bus. Sam was left behind. The combination of anger and hopelessness on that face was heart-wrenching. We made it through the pizza, cried a little, then talked through the disappointment. Trying to manage the anger and emptiness that comes with FA is real and a never-ending struggle. If the emotional challenges aren't enough, the medical management of FA is daunting. Alex, 27, was diagnosed at 8. Her short life has experienced numerous hospitalizations. The fragility and complexity of this rare disease creates its own obstacles. Years ago, Alex anguished over the decision to have a baclofen pump implanted. Her spasticity was overwhelming and the pain too much. She maxed out on oral doses, but had had so many problems during 2 prior spinal fusions, she was understandably afraid to undergo another surgery with associated risks. For me, it was painful to watch her struggle with such a decision, but I let her decide and stood by to support her. After all, it was her body deteriorating. She decided on the surgery. During the hospital stay, there were problems. It was difficult drilling through her spinal fusion to get a catheter through to her spinal canal. A 1-hour surgery turned into 5 hours and an overnight stay into 2 weeks. I watched in horror as residents just didn't know how to manage her fluid balance. Her labs were off, and they felt IV fluid boluses would help bring her labs up. Because they had kept administering the boluses, which her heart couldn't handle, the fluid became too much. Alex had a horrible drop in blood pressure, and things became critical quickly. The end result was a pulmonary edema, which nearly proved lethal. Luckily, the ability to fight and stand up for what I believe saved her. It was scary and horrifying, and I wish I could say that we've only been through this once, but it's just not the case. I never let myself think of what life would be like without FA. Instead, I focus on hope that there will be a cure. And if we can't find that cure, we can at least find a meaningful treatment to stop the progression. Life would be wonderful if we were able to stop this tireless race against time, sustain life as it is today. I have boxes of vitamins and supplements, constantly tweaking them, hoping the right combination will help a little by possibly stopping the loss of speech or allowing Sam and Alex to be able to feed themselves again. It's the simple things in life that are robbed by this disease, things that allow you to enjoy life as others do. Eating out in public restaurants is a thing of the past for us. There is just no dignity in allowing someone else to feed you as others watch. I take great joy in the wonderful accomplishments of them both, their college degrees, career paths, the laughter, and the kindness they so genuinely display. But I also find loneliness in life with FA. When you face a diagnosis with no cure, most of life you once knew disappears. Family and old friends just can't bear the burden of FA. They fade away. My saddest memory is that day that I realized our home no longer embraced the comforting sounds of footsteps of my children. Thank you.
Thank you again to our panel for those very personal and important comments. We're now going to move to our topic 2 polling questions on approaches to treating FA. Again, if you need, if you're running low on battery and need a charger, just raise your hand and someone will bring by a charging bank. So our first question for you today is, what medications or supplements do you take now to treat symptoms of FA? Select all that apply. So our options here are A, itibenone or CoQ10, B, vitamin E, C, vitamin B3, or nicotinamide or niacin, D, other supplements or vitamins not listed, E, muscle relaxants, F, pain medications such as Neurontin or gabapentin, Cymbalta, Lyrica, or opioids, heart medications such as beta blockers, ACE inhibitors, calcium channel blockers, diuretics, H, diabetes medications, I, antidepressants or anti-anxiety medications, J, experimental medications as part of a clinical trial, K, other, or L, nothing. So again, please select all that apply, all of the medications or supplements that you take now. Give you a couple more seconds to get in your responses. Okay, so it looks like the most utilized medication or supplement is itibenone or CoQ10. Followed by that, we have vitamin E and then other supplements. And then it looks like we have a good range of these other medications or supplements that are used. About 5, sorry, that's not an exact percent, but there are a number of you that have used experimental medications that are not part of a clinical trial. A number of you have used other medications or supplements, and some small number of you are not doing anything currently, and we can explore some of those reasons for, why that is when we get to the audience discussion. So our next question for you is, what are you currently doing to help manage FA or FA symptoms? Select all that apply, and you'll notice these are things outside of medications and supplements. So A is physical therapy, including aqua or hippotherapy. B is stretching. C is exercise. This could be either cardio or strength training. D, bracing, such as back brace for scoliosis or leg or foot braces. E is occupational therapy. F is speech therapy. G is modifications or accommodations within your at work, in school, or at home. H is mental health services. I is use of adaptive devices. J, choice of diet. K, other. And L, nothing. So please select all of these things that you are doing to help manage your FA or FA symptoms. So it looks like the most frequently utilized thing to manage FA or the symptoms is exercise, closely followed by physical therapy, stretching, modifications and accommodations, and then adaptive devices. All of the other different types of approaches to treatment have been selected by some number of you. Others have selected, a few of you have selected other. And it looks like nobody selected that they're doing nothing, none of these. This question is, have you undergone surgery to treat or manage symptoms of FA? And again, please select all that apply. A, for spinal fusion or placement of rods for scoliosis. B, tendon release or surgery on feet and ankles. C, cardiac surgery such as ablation or implanted cardiac assist device such as an ICD or pacemaker. D, placement of a baclofen pump. E, other surgery. Or F, no surgery. Please select all that apply. Okay, so it looks like for the majority of you, you've done no surgery. Of those that have done surgery, it looks like spinal fusion is the most common surgery or placement of rods, that is, as well as other surgeries have all been used by some degree by our respondents. Okay. So the next question is, how much do you use medications, surgeries, or lifestyle changes used to improve your quality of life? Or how much do these things improve your quality of life? A is there's been no benefit. B, they've helped somewhat. C, they've helped a lot. D, there's been significant benefit. And E, you're not sure what the benefit has been. So here, just select one. It looks like about half of you say that the various medications and other approaches to treatment that you've used have helped somewhat. We'll explore exactly what you mean by that when we go to the audience discussion. And it looks like about a fifth to a quarter of you say that these things have helped a lot. About 10% have said that there's been significant benefit. 6% say no benefit. And then about 15% of you say that you're not sure if there's been benefit. And again, we'll explore some of what this means. So our next question is, which outcomes would be meaningful to you for a possible drug treatment? And this is to select all that apply. So stopping or slowing of progression is A, B is gain in function, C is to prolong life, and D is other. So for slowing or stopping of progression, this would be even if there is no gain in function, so our symptoms don't get worse. Whereas a gain in function would be a gain in things like energy, strength, mobility, dexterity, cardiac function, or speech. So the highest rated outcome by our respondents is the slowing or stopping of progression. After that, we'd like to see a gain in function, and then in ranking, number 3 would be prolonged life, and then some of you said other. So we have a similar question here. We're saying which of these outcomes would be most important for a possible FA treatment if you were only able to choose one. So here, again, A is slowing or stopping of progression, B is gain in function, C is prolonged life, and D is other. So rank which would be most important to you for a possible FA treatment. So it looks like a little over 3/4 of you, a little over 75% of you, would prioritize the slowing or stopping of progression. I think we heard some of that in our panel about the importance of being able to maintain things as they are. About 1/5 of you would want to see a gain in function over these other things. And then it looks like either maybe one or no people said the prolonging life, and nobody said other. Okay, so this question is, which ability or symptom would you rank as most important for a possible drug treatment today? And you can select up to 3. And I'll read these since it might be a little hard to see. A is improved balance, walking, or fewer falls. B is improved arm or hand function or manual dexterity. C is reduced fatigue. Oh, actually looks like, no. C is improved, sorry about that, arm and hand function or dexterity. And D is reduced fatigue. E is fewer diabetic issues. F is reduced scoliosis. G is improved cardiac symptoms. H is improved choking and or swallowing. I is improved vision. J would be improved hearing. K, improved speech. L, improved urinary or bowel issues. M, reduced spasticity. Or N, other. And choose your top 3. Okay, so here the results are still trickling in, but it looks like the top 3 or so abilities that would be most important would be improved balance or ability to stand, improved walking or fewer falls, and reduced fatigue. After that, it looks like improved arm and hand function or dexterity and improved cardiac symptoms. All right. Our final question here is, which of the following would influence your decision to use a new treatment or take part in a trial? We're going to ask that you select all that apply. A is significant chance of serious side effects, such as cardiac or kidney issues. B would be common mild side effects of the treatment, such as nausea, loss of appetite, or headache. C is the way that the treatment is administered, for example, orally, intravenously, subcutaneous, or an injection into the spinal cord. D is how the time for that the treatment takes. E is other administration concerns, such as radiation. F is changes to the treatment or management plan, such as stopping a medication or supplement or stopping exercise. G is the cost. H is the travel. I is something else, other. And J is none of these would influence your decision. Again, select all that apply. Okay, so it looks like overwhelmingly the main thing that would influence your decision to use a new treatment is the significant chance of serious side effects. After that would be cost, followed by the route of administration or travel. Okay, so with that, we will transition into our audience discussion again. So again, the same thing applies. Just raise your hand and someone will bring you the mic. And again, please state your name before you give your statement, so that way we can keep track of you for our purposes of summarizing the meeting. I'd like to start off our discussion. We've talked, the panel and the polling questions have talked about a number of ways that you've managed your symptoms, but we want to know a little bit about your decision-making strategy for deciding how to use those different approaches to managing your symptoms. So I guess the question would be, whatever intervention that you want to talk about, whether it's been something that's been particularly helpful, something that you've tried that has not been helpful, tell us why you did it, and tell us why you did it at that period of time. So at whatever point in progression or whatever symptoms you were experiencing, What made you decide at that point in time in your progression, or at that point in your experience, you chose to use that particular approach to treatment? So we'll start right here.
Hi, my name is William Stedman again, and I use Dragon Naturally Speaking speech-to-text software. And I found it very helpful because it was getting more and more difficult for me to type, and it takes forever. And what works well with Dragon is that I could type a little bit, so I can speak most of it to Dragon, but it obviously makes mistakes, particularly with my speech. And I am able to change those relatively quickly, so I can put in a whole bunch of text, and I know I've got to go back and edit it, but that really saves me a lot. That's something I've found very helpful.
Sure. And when you started using it, what made you decide to use it at that point in time?
At that point in time, it was ironically after my dissertation, which I slugged through with my own hands typing, and took a very long time. And then when I was working, I realized that you had to say a lot of nonsense. Whenever you write an official letter, you have to put in a lot of buffers and a lot of nice stuff to it. Whereas I'd gotten used to just the facts. So to be able to add all that extra fluff, I didn't want to bother typing it, and I just couldn't. So I had to figure a way to make that more possible.
Sure. Thank you.
Worked out very well.
Yes. We have, we'll just pass it right on down.
Okay. She's right here. So
Hi, I'm Christy Battaglia. I'm 31. I was diagnosed 2 years ago. June 18th will actually be my FA second birthday. Even before I was officially diagnosed, I started physical therapy, occupational therapy, and speech therapy, just because my doctors knew whatever it was progressive, and the faster you got into, they said, if you don't use it, you lose it. So I started all those. And my doctor actually at first didn't order occupational therapy because he said it wouldn't help me, because once you've lost your fine motor skills, there's nothing you can do to get them back. But I convinced him to do it. They had said it's not going to hurt anything. And for me personally, my declining fine motor skills are probably in my opinion, my most frustrating symptom. They're also the quickest progressing symptom for me. And occupational therapy didn't seem to help in the fact that I wasn't regaining anything, but it did teach me a lot of coping skills, an easier way to put on mascara, which makeup for all of us girls is not fun. But when I started on my own, I started building Lego sets. The first one I ever did was 78 pieces. It took me 2.5 hours to put together. It was the most frustrating thing possible. But at the end of it, I had something to look at that showed me how hard I had worked on it. Since that first set, my apartment is now filled with Legos. It's great though, but also Legos, part of what Lego actually does is measure and monitor developing fine motor skills for how a child should normally develop. Part of it is frustrating because of the small pieces, but also I remember building one set that I could not put together. And the age on the box was recommended for kids 4 to 12. And I'm sitting there, I'm like, I'm 31 and I can't even do this, it's ridiculous. But like I said, at the end, even with the most frustrating sets that have the most small pieces, having that to look at to remind myself the process that I went through is rewarding for me. And my goal, ultimately, I am going to get Lego to partner with Vera to give all of us Legos.
Yeah. Oh, no to add next time. Legos to the questions. Yep.
Looks like we might have a new clinical outcome measure,
There
Lego
we go.
assembly. Right.
All right, I hope everyone in industry and at FDA heard that. So, yes, in the back.
Hi, my name is Beth Haynes, and I was diagnosed with Friedreich's ataxia 6 years ago at the age of 30. And to answer your question before I read my prepared response, my reason to participate in anything is my 7-year-old daughter. She was diagnosed with cancer at 14 months old, which was 3 months after my diagnosis. So everything I do is for her. But on a good note, she is 5 years cancer-free. So.
Oh, congratulations! That's wonderful.
Thank you. I have been in 2 clinical trials. The first, EPI 743, lasted for 2 years. After 1 year, my neurological scores improved by 10 points. This significant increase meant I could take a few steps on my own without assistance, whip my walker around in an instant with sufficient control, and I had sustained energy. But I had to refrigerate the medication and take it 3 times a day with milk. This took a lot of planning when I had to leave the house. The second trial, RTA 408, only required 1 dose of medication per day. While on the drug, I could cook dinner, clean up, do laundry, and manage my very active 5-year-old, all without feeling like I had to crash on the couch at 6 p.m. This was huge when my mother typically did all that for me 5 days a week. Riding the recumbent bike at 65 RPMs for 3 minutes was a challenge. Asking someone who had diminished muscle mass, problems with balance, and unsteady knees from pushing herself to keep walking made it extremely difficult to even qualify for the trial. I want to participate in more trials to help find meaningful treatments and ultimately a cure. But I must consider the challenges and the benefits when looking at future opportunities. Thank you.
Thank you. Yeah, I have a question. So you said that you would need to look at the challenges and opportunities for future opportunities. Could you share with us some of the things that you would consider important to you now in considering participating in a clinical trial? Or
Yes, I'm always looking for opportunities for clinical trials. I've been very fortunate to see great benefits in the 2 that I have participated in, and that's always a big hope for me, is anything to improve my ability to be a better mother, a better teacher, and just a better person.
And is there anything about when looking at potential trials that would be important to you to reduce the burden on you?
As far as physical burden, I've watched my baby do a lot of things that most adults don't want to see. So physical things that I would have to endure are not a problem for me. I think the biggest challenge would be I always have to have someone take me where I'm going. I have to rely on other people and the financial means to get there.
Sure. Thank you very much.
Thank you.
All right. I'll go right here in the middle.
Hi, how you doing? My name is Jacob Tompkins. I'm going to stay on subject here and talk a little about trials. I do have some experience doing trials. I've been in participated in 3 clinical trials and 2 studies in FA. This sometimes these trials can have unexpected side effects or unexpected symptoms. Like my first trial was for a simple antioxidant, but it turned my legs from maroon and purple color to flesh toned, which doesn't seem significant for a normal person, but for someone with FA whose legs were always purple, it was nice to have normal color. And also, the last clinical trial I did required me to make 36,000 miles round trip to California. 7 trips in 6 weeks. This led me to, at first it seemed like a daunting task. I didn't really want to do it, didn't know if I could do it on my own. But I thought in the grand scheme of things, that's all it takes me to do to cure FA. I'll keep doing it.
Oh, thank you, Jacob. Okay. Anyone else that wants to share any of their approaches to treatment? I know I see a hand in the back, and we'll go there right after Nigel.
You want me to get?
Yep,
okay.
go ahead.
So my name is Nigel Lenz. I'm 42 and live in Florida. I've had my first symptom at 11 was scoliosis, and so for 32 years now, live with FA, and obviously walking is something I want back. I did have, you started with asking about surgeries and whatnot. Mm-hmm. I have atrial fibrillation, and for that, like when I first learned that I had that, mm-hmm. I was, my doctor told me about several treatments, drugs available, and I was not used to that. Mm-hmm. So I was extremely happy, and I don't know, about 5 years later, I had to have atrial fibrillation, or I had to have radio frequency ablation because it had gotten so bad, and that improved, and I had to have it again. It improved more. I still take meds, but not. I don't even notice if it goes out of rhythm. Mm-hmm. So I want that for things like speech, which is a big concern. I teach college courses, but I do so online because I want to teach live classes, but I speak slowly and people can't always understand me, so it is difficult, and so I would like something for that. Mm-hmm. I also had difficulty hearing with background noise. One of those examples was a place where I worked, and it would get really busy. Mm-hmm. And when it got busy, I had to answer phone calls, and I couldn't hear the people, and they couldn't understand me. They thought I was drunk, and that was obviously frustrating. So, anyways, I would look for treatments for that beyond the obvious that we've all talked about.
Sure. Okay.
So,
thank you, Nigel. Yep, in the back. Yep.
Hi, my name is Antoinette Menard. I'm 53 years old, and I'm actually unusual. I was diagnosed 11 years ago. My progression has been very slow. I still can walk, but I now have to use a cane on occasion. But one of my frustrations right now is because I am 53, I have not been able to get into any trial. My neurologist has definitely put me forward to try to participate, but many of the pharmaceuticals feel I'm too old. And I kind of look at it as, well, we some of us do get older, and so don't you want to see how the drug is going to work on somebody that's over 50 years old, and also somebody that is still able to walk? It would be nice to have that. So what I have been doing, though, since I was diagnosed, I have been taking idepanone, and I will have to say it has helped. One of the things that I notice quite a bit, particularly if I get off it or I run out, is I have issues with a little bit of bladder control. And when I am taking that regularly, I don't have those issues at all. Also, it does help with fatigue, because that is a major one of, I mean, like everyone said here earlier, if I'm very fatigued, suddenly my ability to walk, to think clearly, I do work full-time, it does impact me. So definitely Devanone helps with that. This current year, though, I have seen the most progression. My neurologist also saw it. And so I've just started taking vitamin B3 for about 3 months. And I've also stepped up my exercise regimen. I take yoga, I practice yoga about 3 to 4 times a week. I do some core exercises, cardio. I do rowing. I work on a rowing machine about 2 to 3 times a week. So I try to keep as active as I can. So what I have noticed over the last 2-3 months of intensity, that I do feel a little bit better. I feel a little bit more alert, and I feel I'm walking a little bit more stable.
Oh.
But I just wanted to share my story, so thank you for listening.
That's fabulous. Thank you. So as we transition to our next speakers, with the remaining time that we have, I do want us to continue to focus on forward-looking, what it is that you would like to see in a future treatment. Obviously, we would all want to see a cure, but we're talking about something short of a cure, setting a goalpost that's maybe a little closer for us to be able to reach. So the question for that is, which of the symptoms do you think are worth treating independent of a treatment for the disease as a whole? Or if you want to alternatively talk about the disease as a whole, what it is that you're looking for in terms of the progression of the disease that would be meaningful for you. So I know we've got a couple of mics lined up, so we'll start on the right and move our way over.
Hi, I'm John Cernasek. My response is actually more so geared towards the questions that were asked here. I currently do a lot to manage my symptoms. The one I want to cannabis. It's a safe and effective and reliable source of relief of so many symptoms. The benefits are wide-ranging. Everything from neuropathic pain relief to muscle relaxing to emotional and mental restoration. Cannabis helps with sleep. Anyone with FA will tell you if you're not well rested, the ataxia is more pronounced. It can reduce dependency on antidepressants, painkillers, and other potentially harmful prescription drugs. This information has been widespread for a few years. Medical marijuana has been legalized in 29 states, plus DC, yet there hasn't been an investigation into its benefits with FA. It's not a cure, but we could be using a specifically cultivated strain that targets a specific symptom right now with the doctor's guidance and feeling better. A study would clarify exactly what the benefits are, open the door for further legalization, classification, and accessibility to it in various forms. Until recently, The primary way of ingesting the cannabinoids was smoking. Now this can be done in healthier ways, such as vaporizing, eating foods with it cooked in, even lotion. This stuff can help us. We need to know how. We need a scientific investigation. Thank you.
Thank you. Excuse me, Paul. All right, so I think we have time for a couple more comments. So, Ron, did you have one right here? Yep.
Miss Brenda here.
Miss Brenda?
Hi, my name is Brenda. I live in Canada, and I live on my own. And I have two brothers that live in another province, so they're not really available to help me. And my fear is, my comment is I like stopping a progression, because it's getting harder and harder every day. I need more caregiving, and they do not come at night, and I get into bed myself. So I'm worried that without any treatment, any stop of progression, I'll be bedridden, or I'll have to go into a home.
Yeah. Yeah, no, thank you. It's very important. I think we have a comment over there.
Hi, my name is Jean Walsh. Can you hear me? Good.
We can hear you, Jean.
Sorry. Yeah. So I was diagnosed 35 years ago when I was 19. So I just made you do a math problem. to figure out how old I am. The treatment that I now feel would be the biggest, most meaningful to me is slowing the progression, because as you've heard, having FA is a relentless loss. So it would be so great to wake up in the morning and be like, yesterday I had a bad day, today I'll have a better day, and I didn't lose something. So it would be nice to have the sense of loss minimized. And finally, just to add to that is, what if my 35 years of progression could be 25, I would be standing, not sitting in my wheelchair.
Wow, thank you. So go.
Hi, my name is Kim Welch. I'm 38. I was diagnosed at 19. I kind of wanted to put a face on something that Jen touched on at the beginning. The 4% of FAers who have one GAA repeat and a point mutation. I have the G130V point mutation. My symptoms are a lot different from most other FAers. My speech and hand coordination isn't affected. I have a spastic gait instead of an ataxic gait. So for me, spasticity is one of my main symptoms, and with that comes a lot of fatigue. So I think ideally for a treatment, one would be to reduce either the spasticity or fatigue. But also a lot of the trials are for people with the 2 GAA repeats. So I'd kind of just like to make sure that for future treatments that any potential treatments would also be beneficial for people with point mutations. Thank you.
Thank you. All right, I think we can take one more. All right.
Hi, I'm Sean Anderson. So I've always considered Friedreich's a two-part piece here. I consider it to have the neurological effects and the cardiac effects for me. So in terms of the like the physical, the neurological part of it. See, as I said before, I'm an avid fisherman. So for me, my therapy is tying hooks, because it's real fun to put the line through, and it gets very frustrating at times. But the reward is to see the finished product and hopefully see a fish on the end of it, but it doesn't always happen. The cardiac piece, though, I can't do anything about because that's inside my body. It's not like I can go inside my heart and say, okay, get better, or have a therapy. That I need medication for. So we're very close to a cure. And, right now I feel like I just need to, if I can get the heart done, I can buy myself enough time to get a cure. And it's almost like, I'm watching this, the hourglass of my life, and I'm like, well, if I can just slow it down, and that would be helping out the cardiac piece of Friedreich's, then I might be able to have time to get to the end, which is the cure.
Sure. Thanks, Bill. We'll take the two remaining individuals that currently have mics. We'll start on my left, and then we'll come.
Okay. Of course, the physical and neurological symptoms are the most important, but I'd like to bring up something that has hit home to our family with two with FA over the past 28 years, and that is the financial burden on families for purchasing idebenone or CoQ10 or whatever we've purchased in the amounts that have been considered therapeutic. One day, about 10 years ago, I just sat down and just started thinking how much money we were spending on CoQ10, which we never really knew if it was helping, but as a parent, you're like, well, my gosh, what if I don't give it to them and they do get worse? So you just keep giving it and giving it. And I figured out we probably could have bought a second home. So when you go through FDA trials and you have something that's an approved drug, at least you're in the arena of possible insurance company help and things like that. Whereas when you're in the arena of things that can have been on the market for so long that there are no studies for them that are available to them, then it presents a strain on families emotionally and financially.
No. Very important point. Thank you.
Hi, my name is Teresa Holm, and two of our kids have been diagnosed with Friedreich's ataxia. The thing is, being diagnosed 2 years ago, hope is what we have clung to. As you glance around the room, most of the age of those afflicted are young. This disease is not a once and done. It's drawn out, and we mourn each individual loss. Every day, I wait for the next regression. I know it's coming. As FA parents, we all do. The aged neurologist that diagnosed our kids told us with tears in his eyes because he knew what the future held. Just want to know how we can give the kids more time.
Thank you. All right, one last one, Ron says.
This is Gretchen Anderson again. Just a couple of things. My husband and I were really surprised to see that the cardiac, treatment for cardiac issues was a little bit low on here until we thought about it, or at least until I thought about it. When Sean was diagnosed, what we saw was the ataxia. So our fear was, oh my God, he's going to be in a wheelchair. That's the worst thing. It's the awful thing. His heart is fine. Even when it was hypertrophic, his heart was, it's fine. It's still functioning like how ours are. It's at like a 50-year-old's heart. So that's fine. As long as he stays like that, that's fine. We don't want him to get in a wheelchair. So all our efforts were keep him out of a wheelchair, find a neurologic cure. And what we didn't realize until Sean quickly turned into dilated cardiomyopathy is, yeah, the wheelchair, and went into a wheelchair. Wheelchair is not great, but a wheelchair is okay. The longer I'm in this game, the more I learn that it's the heart that kills them. And it can kill, we had one last year that died at 11. On our group, we have people that die all the time. And I'm pretty tired of having to share in that grief and express my sorrow and know, like we all do, that next time it could be our kid. And so while nobody thinks about the cardiac stuff until your child actually, or you may, but you try to put it off. A cure to give them more time, to keep their heart healthy, to give them more time, is paramount. But along with this, anything that shows some kind of improvement or some kind of slowing or stopping of any deterioration, whether it's swallowing, whether it's hearing, whether it's vision, whether it's walking, whether it's your heart, whether it's the gastrointestinal issues, is beneficial, because even if we can't extend their lives, we can make the life they have here and now much better, a much higher quality of life, and that's important too.
Thank you. So I know we've gone a little over, but I think that's okay. We're all here to hear you and your voices and your experiences, and so I'm glad we had the opportunity to do that. If I could have my academic, industry, and FDA colleagues join me in a round of applause for all of the patients and caregivers that shared their stories. So now we get to move into our concluding remarks. I'm pleased to introduce our FDA speaker for closing remarks, who is Jonathan Goldsmith, Dr. Jonathan Goldsmith, who is the Associate Director for the Rare Diseases Program in the Office of New Drugs within the Center for Drug Evaluation and Research. There, in this role, he facilitates, supports, and accelerates the development of drug and biologic products for the treatments of patients with rare disorders. He does this through coordinating the development of CEDAR policies, procedures, and training, helping enhance science-based approach to rare diseases product development and regulation, and working collaboratively with stakeholders in promoting consistency and innovation in review within the Center for Drugs. He has a long history of working in rare diseases and training in rare diseases, which I won't go into, but join me in welcoming Dr. Jonathan Goldsmith. Do you have slides? Yeah. There you go. You.
Well, so I just want to thank the organizers, the patients, families, members of regulated industry, academia, and government who are gathered here today. This is quite a congregation, to be honest. I've been to many of these meetings and seen lots of them from a distance and close up as well. And you certainly have a very vibrant organization, and you're very focused, I think, on where you're going, and I think you'll get there. That's what counts. So I'm a hematologist, a blood specialist, which doesn't sound like rare diseases, except in hematology, everything we took care of were people with rare diseases. So I came by this over my many decades-long career in academic medicine and elsewhere. I've had an opportunity to work in regulated industry, where I was able to work on clinical trials, safety of products, in vitro diagnostics, and had several drugs that we developed that were approved and are in current use now. So I have some of that perspective as well, and it helps me a lot as a regulator as I work at the FDA to try and see this from all sides of the triangle, if you will. We're now in a transition from the patient-focused drug development meetings that we've held over the last 3+ years to these externally led patient-focused drug development meetings. The patient-focused drug development meetings were set up to support systematic, scientific approaches to methodologies and analyses to bring science to the study of rare diseases and of patient comments, the patient voice. We've learned a lot, and we've also confirmed some things that we knew. Some of us practiced medicine for a long time and knew this already, but patients are uniquely positioned to inform FDA about their disease and its impact. You're the experts. You heard that in the morning from Dr. Bryant. I just want to make sure it's here at the end of the day and confirmed. We have difficulty in FDA over the last decades because the mechanisms to obtain patient input may be limited to a single application that's under review with us. We don't actually talk about the broad field, the disease itself. We have to be focused on that application. But patient-focused drug development is really important to us because in the Office of New Drugs, about more than 40% of our new molecular entities, the most novel, the newest drugs that are approved, are for treatment of rare diseases. So it's really a big focus of the FDA at this time. People think about it, well, rare, not common, but they're where drug development is going, and it's going there full bore from what we can tell. Patient-focused development, drug development meetings, they've also shown us the value of the voice of the patient reports that we've developed at FDA and post on our website. These are helpful to people who do the reviews of the products, as well as they have discussions with regulated industry when there are meetings between FDA and regulated industry. So they've been very helpful to try and focus what might be a really clinically important endpoint. As I've heard today, and you all know, Friedreich's ataxia is a debilitating degenerative neuromuscular disorder. And from this very well-attended meeting today, we've heard quite a few important facts. We've heard about your commitment and your love for your children, your courage and determination as adults, children as teens, to live with your disease and to live well with your disease. We've heard about the heavy burden of the disease for patients and families from your heartfelt descriptions. We heard about the transgenerational impacts of the disease, how this affects families. I heard lots of parents talking today and what it meant and what it means. We heard about the mobility issues and the challenges in terms of activities of daily living that you confront each day. We've heard about the loss of coordination of your arms and legs, including motor dexterity, things that most of us take for granted, turning a key in a lock. And we've heard that these are progressive. And obviously the rate of progression is not the same in each person or in each family, but they are progressive. And the element of time and thinking about how we need to do intervention sooner rather than later comes home from this progressive perspective. We heard about fatigue, muscle weakness, and muscle pain, including terrible post-operative pain, and how people somehow get through that. But fatigue is obviously a dominant theme. We've heard about complications and benefits from surgery, which many of you have undergone or may undergo. We've heard about managing complicated medical care at home. The videos we saw were striking, as you know. And this includes the management of things like diabetes mellitus, for instance, as well as the general disability that people may have. So it's a complex multi-system disorder. Family impacts, I heard also that include social isolation, mental health issues, neuropsychological complications. I don't think these were addressed in as much depth maybe as they probably really exist, but I heard some really poignant stories about what happens and what goes wrong and what can I do to help my kid, for instance. Losses, as you know, include not having a normal or a near normal life and losing independence. I think that's something that has to be avoided at all costs. If there's an intervention, a therapy, as you voted, I saw today, you want to slow the progression of the disease. You want to maintain your abilities to ambulate, to get around, to take care of yourself. We also heard a fair amount of discussion about serious heart conditions as being a problem themselves, but also the anticipation of these heart conditions, what it could mean for me or my family. I heard one good technological benefit comment during the day about managing aspects of the disease and enhancing communication, and trying to use some of the modern tools that we have based on the computer discoveries that have gone on for the last decades. I want you to know, oh, here, sorry. I heard about your views on treatments and the hope for treatments under development, and I heard about your adventurous approach to taking a lot of these treatments, that a lot of people are willing to engage in clinical trials. They'll travel distances. They're worried about cost, I see, but generally clinical trials are paid for by other mechanisms from the drug sponsor, so that may not be as big an issue. But that your enthusiasm for participating in trials was important. And I also heard about your comments about the lack of treatments development in pediatrics. If the onset of the disease is between 5 and 15 years, then a pediatric approach is necessary. And FDA will probably require this as part of the drug development process, I can tell you. We don't let this go by the board. We think that the youngest patients have to be served as well. So just to conclude these very brief remarks, your voice helps FDA as we perform our public health mission and as we evaluate and approve new drugs and biologic applications. I want to thank you very much for including me and the FDA, many of the people who are here, in your excellent program and fostering the strong sense of collaboration that is helping to bring new therapies for Friedreich's ataxia to patients and families. Thank you very much.
Thank you to all our attendees from FDA, especially Drs. Bryan and Goldsmith. We appreciated the opportunity for this patient-focused drug development meeting and are most encouraged and appreciative of your sincere interest as demonstrated by the great attendance and attention we received today. To our pharma, biotech, and academic participants, thank you for your partnership and your commitment to developing treatments. A special thanks to James and my colleagues from MDA and NAF for helping make this possible, but especially I really need to recognize the FARA staff. This was a full team effort. This is the largest gathering of FA patient families to date. As I call each of you, you need to stand up. Jane? Jane Larkindale is our scientific director, and she wrote the letter of intent to make this meeting happen. Jamie handled every aspect of logistics, from the hotel to food to transportation, AV. Felicia, Katie, Kyle, Anne, you guys did everything and anything we frantically threw at you over the last several months, even when you were brand new and didn't even know what you were doing. So thank you all very, very much. I'm just truly honored to work with this team. This really was a full court press for us. I'm most grateful to all the panelists and patient families and FARA ambassadors who have participated and shared so many personal experiences and valuable insights. I've been working in this disease for 20 years, and I'm still learning. And I recognize that this is difficult and different than most meetings and FARA gatherings that we host. And we don't often focus on the challenges and the difficulties. And this really was a challenge for all of us. And I appreciate you being so open and willing to share. The voice of the patient report is incredibly important. And Jane and Katie and I will be working on that over the next several months. And that will be filed with FDA. If you felt like you didn't have a chance to say what you wanted to say, or a microphone isn't your preferred method, or you're online, please write your testimony or your comments to us. Those will be included as well in that Voice of the Patient report. You can submit them to us by email, any of us, info@curefa.org. Email any of us at FARA over the next 30 days, and we will make sure that those are included. My final thoughts, I believe we accomplished the goals we set out to accomplish today. I believe the symptoms and the complexity of FA and the impact on activities of daily living have been discussed fully. We've talked about the current management and the shortfalls, and shared thoughts on what meaningful treatment would be to our daily activities. And we've put the patient voice front and center. We all understand the severity of FA and the high unmet medical need and the basis for urgency and accelerated therapeutic advancement. With this, we know that you are on our side in success and in changing lives. Thank you. And you are free to go to lunch. We're going to have lunch in the back. We are going to convene again at 1:30 with a research update. And so we have several pharmaceutical companies and research representatives here who are going to share some of our pipeline advances and research updates with you in the afternoon. Thank you.
like that. Do you want me to pull it up and then you take? Oh.
All right, we're going to try and get the afternoon session started. So if everyone could come in and get a seat. We'll start in about 30 seconds. So we appreciated the fact that so many families had traveled to be here today for this morning's meeting and wanted to be able to also provide a research update. But I realized that this morning was long and emotional. And so, you know, we're not going to go very long this afternoon. We'll keep this afternoon session to about 2 hours, which is a challenge even after lunch. So this will be a brief update. It's not meant to be exhaustive in terms of everything that's going on. And but we do have several of our pharmaceutical partners here today who graciously volunteered to give research updates on their programs. We will have additional conferences this fall. The Energy Ball Symposium will be September 14th in Tampa, Florida, and that's always broadcast live. And then we have the Children's Hospital of Philadelphia FA Symposium on October 16th. Outside Philadelphia, and that's a one-day-long seminar where we're able to kind of cover more ground time-wise. So there will be more updates later this year and more opportunities to discuss research and the work that's going on. So let me get started. Just in terms of what our agenda is for this afternoon, I'm going to go through just some selected research updates, things that I thought were new that maybe not everyone was aware of. I can't cover everything, and I apologize in advance. If we have time later and there's questions, I will take them. But we do have Jupiter Orphan Therapeutics here. They're going to give an update. Retrotope's going to be giving us an update. We have a researcher from Pfizer who wants to talk about new technologies and how we apply them to the work we're going to do in future clinical trials. And then we're going to have an update from Dr. Lynch on the Moxy or Reata study. So I showed this slide this morning. And it's just a reminder of all the different things that we're trying to do in terms of, you know, funding basic science and drug discovery, trying to put the clinical research infrastructure in place to facilitate clinical trials, things like our biomarkers. We initiated a full court press on biomarkers about 2.5 years ago. And we've been funding a lot of research in this area. Many of you have participated in a lot of these studies. We have our next scientific conference, September 27th to the 30th of this year, and we'll have a lot of updates on our biomarker studies at that meeting. And, you know, I think this morning's session really focused on what we're doing on the advocacy front and working with FDA. It's a quick overview just of, you know, where FARA grant funds have gone in the last 5 years and sort of the general research buckets, kind of as we call them. So drug discovery, gene and stem cell therapy, what I call lead candidates, kind of things that have gone beyond drug discovery but aren't yet in clinical trials. Really basic science, mechanisms of disease, cell and animal models, our natural history. As I said, you know, you can see the bigger investment in biomarker research over the last few years, and then cardiac research. These aren't perfect buckets. There is some overlap. Sometimes I don't know which one to put a specific grant in, and I ultimately have to make a decision. So it's not perfect. And for 2017, we're still in the process of making decision on new grant awards, and so this isn't a full picture for this year. This will change as we go through the year. I'm not going to be able to go through every bar in the pipeline, but you're going to hear about 3 of the specific programs, and I thought I'd just go through a few others where, like I said, I thought we had something to talk about. So at the beginning, or at the end of 2016, we had the results from the phase 3 ACT-Immune study, and you heard several people this morning talk about their experience participating in the study. So the first part of the study was called 301, and that was the 6-month double-blind placebo-controlled portion of the trial, and those were the results that were released at the end of the year that were negative, and that there wasn't a significant difference in the endpoints that were measured between the placebo and those in the treatment group. There were additional aspects to the study. So after the 6-month placebo-controlled portion of the study, there was an open label extension. There were actually 2 of them, and they were called 302 and 303. So what's happened since the end of the year is that those, we've continued to look at the results of the 301 study. The clinical research visits for 302 and 303 have now been finished. So everybody who was in the open label extension studies have now finished all their visits. Their data has been collected. They are in the process of sort of finalizing and doing a process that's called locking the database. And then once the database is locked, we'll be able to look at the additional data from 302 and 303, which gives us a much longer view because those studies collected data for an additional 12 to 18 months in a lot of individuals. So while the top line data wasn't what we'd hoped it would be, we are still continuing to analyze data from that research study and will be into the fall. And so we'll hopefully have more insights. I think everyone heard that there were some folks who felt that they had benefit and enjoyed benefit when they were in the study or in the open label portion, and we're trying to understand that now, why the results didn't coincide with what people had anticipated and what they thought was going on during the study. So Chondrial Therapeutics announced earlier this year that they secured funding, licensing, leadership team, and is really off to the races with a program that started a long time ago. Many of you might know Dr. Mark Payne. He's a cardiologist from Indiana University. He's been doing FA research and providing clinical care for a long time. He's been working on developing a protein replacement approach using something called frataxin. And so Chondrial is now able to take the work that he's been doing in his academic lab and really try and move it forward to clinical program. And so they're now actively working on all the IND enabling studies to hopefully move this into a phase 1 study and give it a chance in patients. And there are many people from the Chondrial team who were here the last 2 days, and I'm thankful for them. And they're really growing commitment. I mean, in a short amount of time, all these folks were new to the FA space, and they've really come on board, and they share our sense of urgency, and I really appreciate that. Epicatechin. Took me a while to figure out how to say this one. Kind of came out of the blue, right? Nobody's heard about this before. So this is a natural compound that mimics a hormone that's made in the body. And it's believed to stimulate mitochondrial biogenesis, so the making of new mitochondria. And if you can make more mitochondria, then you might be able to have improved muscle function. And so that's sort of the premise here. And epicatechin has been in a few clinical trials for a form of muscular dystrophy. And one of our investigators at Mayo Clinic, Dr. Relitsa Gavrilova, is working with Cardero Therapeutics to do a pilot study of epicatechin in patients with FA. They are looking for 10 subjects, ages 10 to 15 years. One of the challenging criteria is that these need to be relatively newly diagnosed individuals, less than 7 years, and ambulatory. The treatment phase of the trial is 6 months, and they are looking at both neurologic and cardiac outcome measures. They've enrolled about 7 individuals, and they're still looking for about 2 or 3 patients. So, Crestor, because I can't say the other name at all. I really tried. Dr. Ian Blair at the University of Pennsylvania, part of the FA Center of Excellence, has been working on biomarkers and looking at different levels of protein in the serum. And he discovered, kind of serendipitously, that individuals with FA have low levels of a protein called ApoA1. And ApoA1 is important for lipid metabolism. And he went a little bit further and looked at other proteins in lipid metabolism, specifically HDL, and HDL was also lower. And that makes sense because ApoA1 is actually a component of HDL as well. We don't know why this is happening, but there have been some recent reports in some of the other animal models of FA, like the fly model, that they do observe lipid metabolism abnormality in the cells, in the neurons, in those models. So this is not completely as crazy as it might sound, kind of out of the blue. Crestor is a drug that's been around for a long time, used to treat cholesterol. Is there anybody besides Dave Lynch who's on Crestor? That was an easy one, Dave. So Crestor is known to work by increasing HDL and ApoA1. And so through the Center of Excellence, Dr. Lynch worked with Dr. Blair, and they decided, let's do a biomarker study. Let's see if we give people Crestor, can we elevate ApoA1 levels? And so this is an exploratory biomarker study, 10 people, 3 months of treatment, and see if these levels come up or not. And then if they do, and we'll try and figure out why, and then it would mean it was probably worthwhile to go further and investigate whether or not this leads to some sort of clinical benefit. All the patients are enrolled. Thank you. I know several of you are here. Thank you very much. The treatment phase is still ongoing, but we expect it to be finished this summer, and hopefully we'll have results by the time of the October research conference. Methylprednisone. So this comes about from 2 different directions. One is that been a number of individuals with FA who've been diagnosed with other diseases. And those other conditions have required the use of steroid treatment. And it turned out that when they were being treated for their other disease, their FA symptoms improved. And improved enough that they reported this. And so over time, it started to become a pattern of hearing this story that when people were given steroids, some of their FA symptoms improved. At the same time, there's been a growing body of evidence that inflammation may be a part of FA, and that's something we didn't really appreciate early on. And it might even play a role in the cardiomyopathy of FA. And steroids are a commonly used drug, especially in conditions where inflammation is a component. So this is another exploratory study where they're looking to see if methylprednisone over 6 months improves some of the neurologic function, specifically walking, in people with FA. Again, small 10 subjects. All 10 have been enrolled, and we're hoping to have the study complete by December of 2017. All right, this one is really going to be a test for me. So Takeda is developing a drug called TAK-831 as a potential treatment for ataxia. Now, so I don't have a pointer. Darn. NMDA receptors. Okay, it's taken me 10 years to be able to say that, and it's the 10 years I've been working with Dr. Lynch. If anybody, a good Trivial Pursuit question, what does Dr. Lynch work on in the research lab? It's NMDA receptors. In the first 10 years I worked with him, I had no clue what the heck they were. But I'm quickly learning. So NMDA receptors are in nerve cells. And these receptors need to be activated for one nerve cell to communicate information to the next nerve cell. And these receptors are activated by other proteins and enzymes in the cell, glutamate and D-serine. All right? And activation's important because if that receptor isn't activated, those necessary information and chemicals can't be communicated to the next nerve cell. And so these, glutamate, D-serine are synthesized in the brain. And one thought is that if we could augment the levels of D-serine in the brain, that maybe those nerve cells that are maybe struggling for other reasons related to FA or other reasons for ataxia might benefit from having an additional amount of D-serine around to help those receptors do their job and allow the neurons to communicate. So this would be a symptomatic kind of therapy if it were to work. It's not attacking frataxin, it's not dealing with the root cause of the disease, but it is working specifically within the brain, in the region of the brain where we know there's trouble with neurons communicating with one another. All right. So I did pretty good. Those were my main research updates, things that I thought were new to share. I can take some questions if we have any. Paul. So the question from Mr. Conance was, how long will we keep EPI 743 on the treatment pipeline? So EPI 743 is up towards the top, and the clinical trial was completed a while ago. I'm keeping it there until the results are out. I'm keeping it there until the results are published. So I think it's important. Even though we've had some brief reports that the results from the study were negative, I think that it's important to have a full published report, and so I think it's important to keep it there until then. And other questions? Yes, Larry. Oh. Will the pipeline be on the website or the presentation from today? Both. So the pipeline is on the website. I updated it a few months ago. It hasn't changed too much. I try and make updates a few times a year. And yes, this portion of our meeting, this research update, is being broadcast live. And we will, because it's being broadcast live, it'll be on the YouTube page, FARA's YouTube channel afterwards, as will this morning's session. So if you have people who didn't get to be here or participate, who would like to watch it, it will be on the FARA YouTube channel. Yes? Sure. The question was about cannabis treatment and whether, you know, what's appropriate to discuss about that. I don't know that we have a full amount of time to kind of go through that, but I think John made a great comment this morning when he was talking about it. I think he was very specific about the benefits that he felt that he was achieving or receiving from that, and the need for treatment trials, right, for further investigation into how or why that might be working. And I think that that's something that, you know, we can take back as the research community and think about seriously, and, you know, what kind of trials could be meaningful and achievable. So from that context, yeah. All right. Well, I would like to introduce our next speaker. And so our next speaker is going to be Chris Rosen. And Chris is the CEO of Jupiter Orphan Therapeutics, and very much appreciate his being here today to give you an update on their program in a special version of resveratrol.
Well, one way with resveratrol, and I don't know if you know where it comes from, but it comes from red wine. So you can choose to drink like 20 bottles a day and you will not need our medication, you know. So that's an option, you know, you can start right now. You know. So I started this company about 2 years ago because a friend of mine was a professor in Miami and he was early with Pfizer and AstraZeneca. He said, we need a new drug delivery technology for resveratrol. It's very, very good in many different indications, but as soon as you go to the level of efficacy, then you get stomach or GI side effects. So since I was dabbling in my earlier company with drug delivery technologies, he thought I could fix that. So, and then I looked at big pharma companies. One company actually had bought another one for $700 million for resveratrol, but they didn't like that it's a natural compound, so they tried to modify the molecule. And stupid and stubborn as I am, I thought that I could fix it, and I have my chief scientific officer, Marshall Hayward. He's also good at those things, and it looks that we are actually on a very good way of doing that. So we call our product Jotrol. And Jotrol is still resveratrol, but very unique, patented, we have applied for patent, delivery system that dramatically increases the amount of resveratrol that end up in blood plasma. And the whole reason for the background to it is that there are a lot of existing human studies with resveratrol that all shows that at a high level of dose on a daily basis, like 3, 4, 5 grams per day, you have an effect. But as soon as you go over 2 grams per day, you start with the GI side effects, which include abdominal pain and diarrhea. And you have problems enough, you know, with moving around, but having a diarrhea and have to go to the bathroom real quickly can cause a little issues, you know. So the whole emphasis now is to be able to bring in enough resveratrol into blood plasma without causing that GI side effect. And that's what we, through mice, rat, and also combination looking at human studies, we believe that we have accomplished. So what we have, if you can maybe understand it, this is the bioavailability amount of resveratrol we get in blood compared with the best available micronized resveratrol that you can buy at nutritional stores today. And we are, you know, 7 to 10 times higher. And also, all the scientists in my program, they also believe that this is a switch-on effect. and not necessarily an area under the curve. So this very high, quick Cmax, it just triggers a switch that then makes the genes and everything works properly. Oops, did I press the wrong button? Yeah. And so among the things that we also looked at, because resveratrol does penetrate the blood-brain barrier. So we looked at IDUA activities and also frataxins in mRNA, and compare with actually our Jotrol formulation without the resveratrol. And in all cases, we can see that there's a quite dramatic increase with our Jotrol dose. These are done with the equivalent of 50 milligrams per kilogram resveratrol in the Jotrol formulation. So why is Jotrol for Friedreich's ataxia? And actually, Jen, you need to also add our program in the mitochondrial area. And as you know, that mitochondria today plays a big role, and there's new papers out on mitochondria and how important that is. And we have then seen that resveratrol upregulates frataxin gene expression in both human cells and animal models. And it induces mitochondria as like a nutrient for mitochondria. And then what really encouraged us and why we started with FA very early, it's one other program we're doing as well, but FA is one of the two indications that we work on here. And that was what Martin Della Tucci study done in Australia, and where he actually, on the paper he presented, there was kind of a failure because his primary endpoint was increased frataxin levels, but he only measured it once at one time point, and we think he actually missed it. But then there's a lot of other areas in this study that showed that a lot of things that you've been talking about in this meeting here, the modified FAR scores, speech, hearing, and so on, was improved quite dramatically in the study in Australia that was done. And this was an open label study, so he first tried the same patients on 1 gram per day, and that really gave no difference. He increased it to 5 grams per day with associated GI side effect, but then you can see that he had quite 4 out of 5 of the endpoints were met. And so this is really our basis now when we're going to file our own IND here with FDA and go to say, okay, as long as the FDA then will accept those endpoints, we feel that we can have a possibility to have a product in the market relatively quickly. So the study we're doing is funded already by the Australian government because of the encouraging result that was on the first study. And as I said, we're going to have about this. I mean, FDA, of course, we're going to file an IND, and FDA might have some other additional things in there, and we, of course, have to abide to the rules. But maybe we're going to try with, in the label, to state that the capsule, by the way, is going to be oral gel capsules. And I would suggest to FDA said it can be taken with either a glass of water or a glass with red wine. So we'll see how that flies. We want to complete about 40 subjects, 24 weeks of exposure, and with a crossover design. So this is not just exploratory. This is a study where we hope we can get actually, with FDA's blessing again, accelerated approval and have a chance to get it maybe on a conditional approval, get it on the market even after this study. And the PI, of course, is Martin Della Tucci, that quite a few of you know already really well. It's hard to forget someone better than him, since Dave Lynch is so busy with all the other stuff that he's doing. But we hope to have him getting his blessing as well. So this is where we are today. And if you think about I mean, any pharma people around, they know that this is like really gone fast. We started a company June 8th, 2015, so it's soon second anniversary. We have already now, it's actually in the process of manufacturing clinical trials, and that is done by Catalent in St. Petersburg, Florida, which is close to a few people around as well. We are submitting orphan drug designation in June. Actually, our regulatory expert sits down there, and is it June? Yeah, thumbs up, yeah. And of course, as you know, it's no problem to get an orphan drug designation because of the FA population, but we expect that to be done fairly quickly. We expect our PK study that will be done actually by Inventive Health in Canada to be completed in the third quarter. As far as the study, the complete study results probably not going to be until another 3 months later. But in combination around that time, we'll also expect to do the IND for FA. We're very aggressive with our plan because we're small. We wanted to go fast. We hope to be able to start the phase 2 study in by the end of Q4. The Australians are a little easier to deal, so we don't know how, but we want to see that we have FDA's blessing. So it might be that will end up being Q1 2018 that the start is. Top line results, the first half of 2019, and then it's a question of can we get an accelerated approval or not by FDA and so on. But that's what we really targeting, because we want this product to be out, have a good chance to being out in early 2020. And if the result will be the same as the open label study, you will definitely see benefits of this product. So that's our gamble, and it's our own gamble. We put our own money into it, and so on. So it's we really look forward. I mean, when you meet with you here, and as we met in Tampa in September, I mean, you just want to have this drug out tomorrow. I mean, that's how the feeling of it is. So we're going to do our best to do it. Thank you very much. Jen thought that I could answer questions. That I always refer that to Marshall. Yeah. Give Marshall the microphone. Yeah, and then. You want me to repeat? Yes.
Could you repeat the question so we can hear it in the back?
So the question was, is the increase in frataxin thought to be related to the increase in mitochondria from the mitochondrial biogenesis effect of the drug, or is the increase in frataxin something more direct on the frataxin gene, like the frataxin promoter?
The expected outcome is that there's enhanced gene expression and frataxin accumulation, which would then find its way into overall energy metabolism. We've seen increases in the messenger RNA presence.
Yeah. I mean, it's
Yeah.
Kyle?
This drug, it sounds like that it might be applicable to other diseases as well.
Yes.
Are you pursuing any other diseases in parallel, or are you focusing all on FA right now?
No, we are in parallel. We're doing MPS1, multipolysaccharidosis 1, which is a lysosomal storage disease disorder, and that's actually how we got funded originally. So we cannot say, oh yeah, we don't. It's only 1,000 of you, and there's like 5,000 of them. So we do that in parallel, but it's the same product. We estimate it to be the same dosing for both indications. So it's not. So and it has possibility also within other ataxias down the road. I mean, for example, it's University of Cumbria in Portugal contacted us for Mercader-Joseph disease and so on. So it's been quite an interesting ride, this, because first scientists didn't want this to be resveratrol be effective because of some history. Now all the scientists are coming to us and say we want to be involved. So we have some very, very great scientists that is supporting us and so on. And I'm not a scientist. I'm a passionate, crazy guy that when I see something I think I can fix, then I go for it. That's
Thank you for
yeah.
you can fix that because we all believe in you.
Okay, thank you, Kyle. Yeah. Any more questions? Yeah, Jen warned me about that, you know, because it's this is not defined yet. I mean, for us, we're a very small company funded primarily by myself. And when Australian government says we fund the clinical, and then with 3 or 4 sites in Australia, we have in our original plans to use at least 1 site in the US, and it's not out of the question yet. And that's we had talked with Emory in Atlanta as a site, and also Jen have said, well, maybe they can. Your fantastic FARA group can assist with travel or whatever. So it's like we
to Australia.
No. Why not? To Sydney and Melbourne, you know, I think that puts some pressure on your patient organization.
Wow.
Yeah.
No more questions for
No.
you.
No, but we of course the discussions with FDA will also guide us in that. So it's not absolutely not out of the question. In fact, we would like to have it as long as our cash can afford it. You know. No? Okay. Thank you very much.
So next we have Dr. Robert Molinari, who's the CEO of Retrotope, and he's going to share some results from their first clinical trial in FA. So Dr. Molinari.
Yep. Thank you, and thank you for the invitation from FARA to present today. This trial, I wanted to first talk about the drug that we're using. So RT001, you've heard about coenzyme Q, now you've heard about resveratrol in wine. So we're having a sort of nutrient theme. We actually work with fats. In fact, linoleic acid is the fat you get in salad dressing and meat and a whole bunch of things. And I want to pose a question, because everybody's always talking about frataxin as the root cause of this disease. And indeed, it is the trigger that starts a whole cascade of things that go wrong, including iron accumulation, dysregulation, and accumulation in mitochondria. But why do the mitochondria fail, and why do the cells die? Our postulate is, without lipid peroxidation, those triggers really don't cause anything. If you can eliminate lipid peroxidation, everybody knows that oxidative stress is a huge problem in Friedreich's. And if you can eliminate that oxidative stress, just perhaps the toxin can be rendered non-toxic. So we all agree with frataxin, but without lipid peroxidation, maybe it doesn't do too much. So it turns out linoleic acid is present in your membranes. It is a legitimate and a part of cardiolipin. Cardiolipin is so named because it's in the mitochondria, and it actually is responsible for energy generation as a polyunsaturated fat in the mitochondria. And it is very susceptible to oxidation right at that position where the H's are sticking out. So one of the oldest tricks in chemistry is if you want to stabilize a bond, any bond, you can make the atoms at either end a heavy isotope. So our rationale to use this is by putting a couple of extra neutrons on that position in deuterium, It's not a radioactive isotope. Forget about that. There's several grams of it running around in your body. By using deuterium, we can actually make that bond something like 12 to 30-fold more stable. The fat is still exactly the same fat. Deuterium is still hydrogen in every chemical sense, except it's a bit more stable. It does not affect the biology of this essential fat at all. What it does do is shut down lipid peroxidation chemistry at that position. And that stops a chain reaction that, if frataxin is the trigger, once you start this chain reaction, a signal triggering event can generate thousands of damaged molecules. And that's what causes the cell death, not the fact that you have the trigger in the first place. We've actually borne this out also in other diseases. Things like Parkinson's has synuclein, extraordinarily toxic to brain cells. In the presence of our drug, it's not toxic to anything. And we've done that and looked at a number of systems. So if we're trying to downregulate oxidative stress, this is one animal model we did. And iso and neuroprostanes are measures of how much oxidative damage is done in cells. And pretty much across the body, the liver, the cortex, neuroprostanes are from fatty acids in the brain. Isoprostanes are from fatty acids elsewhere. You can see that the things labeled D in the wild type or D in the transgenics, in every case has much, much lower amounts of oxidative damage. This is in mouse, but it's also well transferable to human. So we could talk a lot about the mechanism of action here, but I'm just going to go on and say, okay, we decided to move early into human patients, and Friedreich's ataxia is a lipid peroxidation storm. We're trying to downregulate lipid peroxidation. What might happen? So we designed a very early first-in-human study. It was, first of all, the limitations of this work is that it was short, 1 month, and it was small, only 18 patients. But we did realize that there was a huge placebo effect in this disease as measured over and over again by others. So we did a double-blind placebo-controlled study with a 2-to-1 randomization of drug to comparator. And the comparator, by the way, was the same fat that you normally have in your diet, just to prove this is not some non-specific fat effect, but in fact is the result of lipid peroxidation. We also put, by the way, when I say PUFA, that's an acronym for polyunsaturated fatty acids. We put the subjects on a low PUFA diet, 3 to 5 grams a day. If you're camped out at McDonald's, you might eat as much as 15 or 20 grams, and we did not want that as a diluent to our drug. It's not part of our long-term protocol. We can overwhelm pretty much whatever you eat with drug, but in this small, short trial, we wanted to make sure we all had the same background. It was the first in humans, so the primary endpoints are safety, 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 tolerability, and pharmacokinetics. But we also looked at FARS neurological scales, time 25-foot walk, the classic things that are looked at in Friedreich's ataxia. And we also went to school on some of the other people who were doing trials and said, gee, exercise capacity is interesting. If the mitochondria is compromised, what better to measure your total energy output than a peak workload or a VO2 max in an exercise experiment? And in some ways, that is the direct measure of the things like fatigue and other things that what we've heard earlier today are bothering people the most. So we progressed forward. We had even number of male and female, 2 cohorts, 2 doses. To give you an idea, this is like a fish oil pill. It's sort of a nice big gel cap, by the way, made at that same place in Florida where JOT is making their drug. And we had 18 patients enrolled, and we went through the protocol. The one thing that was a little bit of a glitch was we did not block randomize, too small a trial really to do that, and we had much more severe patients in cohort 2 than we did in cohort 1. So when you hear me report results, we really didn't have enough placebo patients in each cohort, only 3 really, to measure the placebo effect. But when you combine them, we had 6 patients on placebo and 12 on drug, and that was much more interesting statistically. So we're going to be looking always at all patients on drug versus all patients on placebo. We also call it comparator. So the first thing I want to say is this is a very, very well-behaved drug. It should be. You eat it every day. And we were able to, at the high dose, get up to 30 to 40% of the plasma fat as the drug. That's a huge amount of uptake. And what it told us is, okay, I know it was the most unpopular part of the trial that you had to be on a diet, so next time we're going to relax the diet. Don't worry, guys. So here's the stunning part. It was placebo-controlled. We actually didn't even calculate changes from baseline because there's a large placebo effect here. But if you look at the peak workload in this very small study, none of which was powered to do this, we got a statistical signal at a very robust level of a p-value of 0.008. The drug patients very substantially outperformed the placebo. And again, this was something like 25% increase in the workload generated. We then went on and looked at the VO2 max. And as you know, that's also a very good measure of your sort of capacity to generate energy. That was a very robust trend and also went up something like 16%. So these are fairly major numbers, and it all occurred in 1 month. And you could say it's variability, but you know the statistics don't lie. The peak workload was, in fact, quite statistically significant even after 1 month. So we were pretty excited by this. We've been fairly conservative about what we've been claiming about this study. I was just talking to someone from FDA here earlier, and he said, well, with this you should apply for breakthrough. We'll have to think about that, but this is a pretty strong result. We then went to FARS Neuro, and I want to point out a couple of things on this slide. It was not statistically significant, but if you look on the left at the placebo or the comparator arm, you'll see that one gigantic outlier who improved some 12 FARS points in the placebo group. This has happened over and over and over again in people who use FARS scales for this disease, and I think Edison had similar problems. There were other problems where these scales are very, very subjective and can be part of a very big placebo effect. The other thing is, if you look at our drug, from baseline of 0, which would be the expected change in 1 month, we had some 5 points improvement in the drug, but in the placebo, we had some 3 points of improvement. So this proves that you really have to look at these things in statistics as not against baseline necessarily, but against placebo. And in that sense, you know, you have these big outliers and the large variability in FARS. I think some of these measures are better than others, but this is a classic example of what happens when the measures are subjective. We all know about the timed 25-foot walk. I think a lot of you have been tortured in various ways trying to do this. And we saw no signal. Now, if you had complete no progression, you would only expect about 1/10 of a second difference in the stopwatch between the beginning and end of the trial if you were just progressing normally. So we said we really didn't expect anything when you're in the clinic with a stopwatch and a clinician running ahead of you trying to measure when you cross the line. That's probably not going to work. So we didn't see anything, but we also had a very new kind of, being in Silicon Valley, new kind of little device which we attached to the, I'm wearing it here, the legs of everybody on the trial. And this little guy will measure within hundredths of a second your position and motion and accelerometry in 9 different dimensions. I still don't know exactly how they get 9-dimensional space, but that's what it does. It's accelerometry, gravitometry, and a whole bunch of other things. As in the true Silicon Valley story, we had some high school students put this together for us. And when we looked at this, we could tell very accurately, as you did the walk, where each footfall occurred, because that's a defined point in position. And when we measured that accurately, in the case of the trial down to 1/10 of a second, now we can do hundredths, we found a very interesting observation, and that is, if you add up everybody's strides and look at how fast they're going, again, stride, this is always reported in Friedreich studies as a reciprocal, so this is your stride speed, the reciprocal of the time. The patients actually on drug moved 7% faster. in 1 month. So we're pretty excited about that. We decided to beef up this technology that's on my wrist, and like on my wrist, and like I said, we can now do hundredths of a second measurements, and that's probably more than we need. But we also outfitted at either end of the 25-foot walk little beams like you have on your garage door openers, so you don't squash your dog when the dog tries to come out. Well, now this actually measures and integrates when you cross the line and can measure the time 25-foot walk down to hundredths of a second. So that's planned for our next trial. What was probably most interesting about this is that the efficacy measures of things that are less well studied in a natural history setting in Friedreich's, like peak workload, VO2 max, were completely correlated to the things that are studied very well, like time 25-foot walk, which you can see has sort of a floor effect if you look at the lower left slide there. If you look at all of these versus FARS Neuro, which is kind of the gold standard for some measure of progression on the neurology scale, these are all very highly correlated. What this says to us is we have a lot of flexibility on endpoints here. One of these is as good as the other. There's an enormous amount of data on FARS Neuro, but it's a very noisy scale, as I showed. Peak workload, VO2 max are not. They're accepted in the cardiovascular division of FDA with kind of 10% change is clinically meaningful to patients. We saw something like 15 to 20%. So we think this is sort of the first step at helping the community understand that these measures, which are very, very reproducible, might be a very good surrogate for overall progression of the disease. And we're presenting some of these at various conferences. So what do we conclude? I'm going to go back. The other thing we can do with this graph is we can do regressions of the lines on this progression of disease. And we can say, gee, if FARS scale goes up 3 points a year on average, how much peak workload is that? How much VO2 max is that? If you actually cross-calibrate it with the time 25-foot walk, you get the exact right answer in terms of the progression of the time 25-foot walk over time. So we can predict, based on these correlations, how much change we have. And essentially what we saw is the study was safe. PK was absolutely spectacular. There is a form of this fat called arachidonic acid that needs to be formed in order to get into the brain. That was formed biologically in your liver, and we actually detected it in the patient's blood, so that's exactly as we expected. It was quite tolerable as a drug. Essentially, it's just a high-dose fish oil kind of experiment. There was some diarrhea at the very, very highest doses, but that was 9 grams a day, which we kind of knew was pushing it. And patients complied very well with the diet. In fact, in some cases, too well, fell off the bottom end of how much of the polyunsaturated fats they were supposed to have. It was an unexpected positive clinical result. As I mentioned, there was a statistical result in workload, and when you do those regressions, each and every one of these corresponded to about a 1 year, and I am using the word reversal, improvement, whatever you want to use, in these trends. And then there was a strong trend in the other 3 measures if you count the electronic time 25-foot walk. So this clearly meets the criteria for us for other development of the endpoint using the development of the drug using CPEX type endpoints. We think that can yield a much shorter trial, something in the neighborhood of 6 months, and we have designed a trial for starting in September to do that. I guess I can say euphemistically, we're seeking clarity from FDA on endpoints because the one thing we don't feel we can use in this disease is some of the things that have been coming forward lately about activities of daily living. That would require trials of thousands, and I don't think we have patients available for that. So I think that's it, and I'm happy to take questions.
So I actually would like to maybe make a clarification. Sure. So Bob, I thought you raised a great point that there's variability in the scales. We do see placebo effect. We've seen this in the idebenone study. It's been reported in the EPI study. And so placebo effect is something that's well known in almost all diseases.
Right.
It's fairly common. Now, what you called the placebo effect with the FARS exam, you suggested was due to subjective nature of the scale. And I argue that because the FARS is actually pretty quantitative. There's most of the elements involve quantitative assessments, timed measurements, and things like that. I think the challenge with that scale has been that there's a practice effect. And if people aren't well practiced in before they come into a trial, there can be more variability because you're just learning how to do all of these measures. And when you do something multiple times, you do tend to get better at it. So one thing that I think we've learned with clinical trials is that people need to get practiced in to some of these measures. And so I think the challenge with some of the outcome measures are things like practice effect and also human nature effort.
Yeah.
And that's something that we have to live with, I think, in clinical trials.
So I'm not going to go back to the slide, but if you recall those FARS results, there was two aspects of it. One is there was a zero line, and all the patients, regardless of placebo or a drug, were well below zero, which means there was some sort of placebo or practice effect for whatever reason. And then there was a spread that was also related to things.
Yeah.
And that's sort of the difference in the two. One is kind of a variability, and I think even in the long term,
We do see, yeah.
Yeah, you do see it. I think the FARS goes up on average about 3 points per year, plus or minus 7 or 8, I think is the number I remember.
Yeah. But I think some of that variability is in part related to just practice and effort and other things. But just wanted to clarify in terms of the activities of daily living is a very subjective scale.
Right.
Whereas something like the FARS is more quantitative.
Absolutely, and I wasn't trying to put the two on the same footing at all. But I did say FARS scales kind of generically, so I apologize for that. Yeah.
Researchers looking at the SCAs in ataxia, and the question we're discussing wearable devices, and it was actually Dr. Schmelman who asked if wearable devices could be placebo resistant.
So that's very interesting. If you noticed on our wearable device data, there was absolutely 0.000 in the placebo population. Patients are asked to walk as fast as they can, and I don't think they have much sense of what it is they're doing with their feet. They're just trying to do that walk as fast as they can. And you're kind of not conscious of how you're moving your feet, and that's sort of reflected in the fact that electronically you see no placebo effect in that number. And then when you have the drug, hopefully that's a drug effect that you see an improvement. But I think I agree with you. The data we haven't even begun to analyze yet is all the sort of stability, the side-to-side motion, the, bit of trivia. We looked at Fourier transforms of all the motion and actually saw some interesting things as well. So you can analyze that data to hell and back, but we right now have just used stride time only. Sure.
Regarding the placebo effect for any of these drug trials, does not knowing what an expected outcome is help that? I mean, I know like the drug trial I did, I had no idea what it was supposed to do, so that I couldn't think something was happening.
So people think drug trials are about psychologically knowing and biasing yourself because of these things, and I don't think that's the case. Quite frankly, just because you're on a drug trial, you feel better about yourself. You're doing something about this disease. You're fighting it. In Parkinson's, that creates something like an 18-month placebo effect because that good feeling about being in a trial actually releases dopamine. So the placebo effect is a combination of lots and lots of things.
Have you I wasn't clear as to what the age range of your initial study was. Part two of the question is, have you considered what the lower end pediatric implication age range would be on your September study?
So we were 18 to 50 in this study, and we actually are asking FDA if we can go into adolescents because the drug is so safe. So we'll see what they say about that.
Thank you, Dr. Molinari. That was encouraging results from the first study. And Sue Hagen, thank you so much for making a nice liaison to our next speaker. So next, we're going to actually talk about wearable devices. And so I have Dr. Mirlov from Pfizer, and come on up. Thank you.
Okay. Thank you, Jan, and everybody for the opportunity to be here. I'm very excited. I'm relatively new to Pfizer after spending several years in the Department of Neurology at Harvard Medical School and then as a professor of pediatrics at Mass General Hospital for Children at Boston, Massachusetts. I had the pleasure to join Pfizer and help with a couple of digital innovation solutions for the rare disease research unit. And that's just to make my legal team happy. Okay. What I would like to talk to you about is actually to hear more from you. For me, after spending some years in research, I realized that a significant value is not a p-value, is actually the patient voice value. I will keep my slides very short, and I would like to really hang the microphone back to you to, so you can educate us of what you would like to be able to do with wearable devices as the trend is growing, and we are interested in getting them more and more integrated into clinical trials. I think what we all need to agree and accept is that health in general goes beyond the four walls of a hospital exam room. And those wearable devices that we put on the wrist or on the foot, they give us the opportunity to collect real life data of activity of daily living that is not biased and is not subjective and it is continuous. And it's kind of surveillance type of data, but this is really what we want to know about patients and how it affects your life. And before I move to the next slide, I want to entertain you all and invite you to join my imagination with regard to the following scenario. Say somebody from the FDA came here this morning at 7:45 a.m. He came into the room, he took a picture. There were maybe 10 people in the room. Based on that picture, Jane did a horrible job organizing this conference because it was very poorly attended, right? The same guy from the FDA came back. The time was 1 o'clock. We were all outside in the sun eating. He took a picture, maybe there were like 20 people in the room. Based on those two pictures, the conclusion is that this wonderful meeting was very poorly attended. The point that I'm trying to make is that snapshot is not the way to go. If you really want to know what's happening, you have to have continuous monitoring of the true behavior of what happened in real life. The clinical grade of wearable devices in the market is going really high because many, many people start to realize that this is a way to go. And this is what you guys might experience in the clinic. You know, you might have patient A who is doing the farce and perform very badly on that day, on June 2nd. And patient B, remain at no change. But that's what happened in the clinic on that day. What really happened at home is that patient A was doing pretty well, and just something happened on that particular day of testing that made him or her to perform poorly. And that's a bias, And that's not a true representation of what happened. And that's a recipe for trials to fail. It's a recipe for conclusions to be wrong, and to come eventually to realize that a drug is not effective or is effective based on data that is really not representative. So here is the concept of activity monitoring in the wild, as we call it, and how we can make it work. So the concept is very simple. There is a device like one of those, for example. You put it on the wrist, you can put it on your arm, you can put it on the foot, somewhere that is comfortable. And it is continuously monitoring some kind of behavior, activity monitoring, if it's a gyroscope, if it's a magnetometer, if it's an accelerometer, if it's a heart rate monitor, EMG signals, conductive skin response, temperature. There are a lot of things that we can measure today, with the size of a coin. You can also add a microphone, which we believe very important in Friedreich's ataxia, because speech is something that is very important in terms of quality of life, and something that we would like our drugs eventually to be able to make a meaningful change. So we want to know, not because we're nosy and we want to know what you're talking, we're interested in the quality of speech and how it changes over time. And then you guys are basically just doing what you guys are doing. You're at home, you're at the park, you're going to see a movie. You do what you're doing, the device is on, and we have Bluetooth and wireless connectivity that basically, whenever you are at home, there is some sort of a data hub, like a smartphone or a telephone or a computer, that the device connects to it. All the data goes immediately to the cloud. You don't need to press any buttons. We have those people in the clouds, the algorithm people, who will use very sophisticated machine learning techniques in order to analyze your behavior and movement pattern. So that's the basic concept. It's just a concept slide. And this is the way we would like to see the future of clinical trial, taking the FARS, for example, and try to objectify and digitalize that functional assessment that is done in the clinic. And I know you guys, you're testing for that exam. People are, they do the finger nose test. Everybody wants to do better. So these obviously are very sensitive to practice effect as well. So here is what we would like to do. We would like to take elements, components from the FARS and look for them, try to hunt them in the wild by using those wearable devices, and look for those things that we do in the clinic, but instead of bringing you to the clinic, we save you the hassle, and instead of having one data point that is a snapshot, we have 200 data points a day of what you guys are actually doing, because you wear it continuously. So we can collect data about gait, we can collect data about how you stand and the balance and the sitting posture, but we can also collect data about computer typing as we learn more and more to understand that this is a very important skill and capacity for communication. So we can have you guys typing things as part of the clinical trial, and we would be able to monitor what you guys are doing. To make it even more complicated, we can look at tremors and accuracy of movement and manual dexterity. And in order to reach that, we would have to make you engage in some video games. Again, at home, you would do it on your own device, but we would be able to get all this information, basically use those activities as proxy of the FAR scale instead of bringing you to the clinic. And we can also go and do things like vibration and sensation. We all know the FARS, the sheen slide. When you have to slide your foot over the sheen, it's kind of not something that people would normally do unless they are told to. Right? And if they do it, that's a different question. So we would have to engage you in some sort of a video game in order to capture that, unlike walking and standing and sitting, which you would do normally. And one of the things that we would like to do, be able to do, is to monitor the progression of your symptoms in real life using those devices, and more importantly, looking at speech. So this is an example of voice recording app that can be considered as a solution for monitoring speech instead of getting you to the clinic every time or every 3 months get a snapshot. You can be prompted potentially on a daily basis to read a sentence on your phone. All the smartphones have a recording device anyway. We just need to make sure the quality is kosher and is suitable. And then, you will be able to hear a sentence and repeat it, or say a sentence, repeat it. Maybe not daily because it sounds like too much, but we can do it weekly and get some level of understanding of how your speech changes throughout the day or throughout the week without getting too much of a burden. So I think this is really the time for you, for the last 10 minutes or so that I have with the microphone, to share your thoughts about what does it make you feel, how would you like to have wearable devices being integrated in clinical trial? What would work for you? What is important for you that we measure? What data is clinically meaningful? How much is too much? You don't want to be overloaded with like 10 wearable devices, right? Do they have to be shower-proof? Do you want to be able to swim with them? Do you want to be able to shower with them? There are a lot of things that come into mind, but it would be a huge mistake for us to start even designing any of this without getting feedback on what could work. So that's why I'm here, and it's time for you guys to talk.
Jamie, you got a bunch of people up here.
Okay, my question is, if you're going to monitor data, I was at a symposium 2 weeks ago. They were very concerned with the amount of data that would be generated and how they would store that. You said it was going to go straight to the cloud. Was this implemented yet, or is this hypothetical?
There are two questions, and thank you for bringing this up. I think we're all afraid of the same. We have this fear of big data analytics and ended up with a huge amount of data that we won't know what to do. So forward thinking is necessary here to put the resources in place and the computational algorithm people in place to make sure that we actually know to interpret this data and make sense out of it, because it is collected in the wild without us being there for visual confirmation. That requires a lot of validation studies to do and pilot studies in order to be able to make sure that we measure what we think we measure. Some of these things are already being developed and in other therapeutic areas. So this is not a fairly new thing, but the idea is to make something that would be specifically geared toward addressing the specific issues that Friedreich's ataxia patients have. That's, I think, something that would be innovative and that we would be very interested in exploring in the near future.
Like, for example, they were talking about if you monitor someone's heart rate, that for say a month, every day, 24 hours a day for a month, that's terabytes worth of data, like huge, incomprehensible numbers.
Right.
Like, where would all that go? Cloud?
Well,
yeah, exactly.
Luckily, the clouds are big enough, but but what I can tell you is that there are some. That's part of the pilot that is needed, but I can tell you that in other therapeutic diseases, for example, Duchenne muscular dystrophy, we realized that 2 weeks of data collection is representative of about 3 months of data collection. You don't need actually to burden the boys wearing a device continuously for 3 months, because the variability is pretty much being achieved within 2 weeks, as long as these are not 2 weeks of summer vacation in the Bahamas as opposed to school days or something. Yeah.
So Amir, thank you for a very stimulating and exciting presentation. You were with us today, so you heard, no doubt, from our patients and their families and from the FDA commentators how important fatigue is in this community. You know, for example, that we've lost what we think when we listened to our families, were positive results of phase 3 clinical trials.
Right.
That because we could not measure fatigue, we think we've lost that, and we have to go back to the drawing board and maybe do that study again. I'm wondering, within your own experience with these wearables and predicting future developments, how you think that wearables might be able to help us measure fatigue and the lack of fatigue back to energy.
I think fatigue is exactly, you're very spot on with that. It's something that we all care about very much. We keep hearing from you guys that it affects your life and can change, making it's a game changer between a good day and a bad day. When you have continuous data, you can see how fatigue kicks in and how you get more tired toward the evening as opposed in the morning. And you could see potentially, if you have a treated patient versus an untreated patient, what is their fatigue curve? How does it look like? Does it make any difference? It's not like just a snapshot of every 3 months. You come to the clinic and you fill out a questionnaire about how tired you are. It's really, it's one way of collecting data, but definitely not an accurate representation. The other thing that those devices, they are sleep with devices. You go to sleep with them. So you get a lot of data about your sleep patterns. If you nap during the day, we would know. If you are just being lazy and watch TV and not being asleep, there is a different kinematic footprint, if you like, on how the data would look like from a page from someone who's just lying on the couch watching TV as opposed to nap. If you nap during the day, it means your night sleep is not good. So even just counting the moments or the time of inactivity periods during the day could be different from one patient to another. Again, you cannot do it unless you remotely collect continuous data.
Hey, thank you. This is just a very nitpicky type thing,
Okay.
but is there any way we can make it in different colors? Because like a black thing around your ankle is bad enough that people think you're drunk, but they think you're drunk and arrested. Like, it's just not a good idea.
No, thank you, and I encourage you not to be shy. Listen, you have no idea how much you're right. I mean, I can show you other slides that I have where actually assorted colors and design and how slick the device is one of the top priority on our list. Of course, accuracy is one thing, but we do want it to be fashionable. We do want to be people, have people be proud to wear with it and not feel another layer of embarrassment, because really that's not the idea. If we can avoid wearing it on the foot, if we can embed it into the shoe, for example, wouldn't it be more cool and will take the embarrassment away? I think so. It would be actually interesting for me to hear if you guys want to share about your challenges or willingness to interact with mobile electronics and being able to control some of these things via your phone, playing video games. Is that something that you would feel comfortable doing, or that would create another, you will not be in compliance because you will not follow what we want you to follow? I mean, there's one thing about just being yourself and do whatever you guys do, which will give us one type of data. But we would also hope to collect more sophisticated data. For example, we want to know exactly how you type and how well you type and how fast you type and how accurate you type. Is that type of a game would be something that you would be willing to experience?
I think that most patients are willing to do whatever you ask of us. So I think that's something you'll find in the FA community. My comment was about, earlier you said, doesn't need to be waterproof, doesn't need to be whatever. For me, I thought, I don't, like, Like if it could be in this bracelet where I never have to take it off, and it's completely, I think the key is for this type of stuff, and you'll probably agree, is that it's totally passive.
Exactly.
Once I put it on, I don't have to think about it at all.
Right.
And so that's my feedback. I mean, yeah, waterproof, you know, I can wear it in the metal detector in the airport. Like, you know, I don't ever have to think about it once.
And
I get
battery
it on.
life, which will relate a lot to what you said, because if you have to charge it every 2 days, then there's always the risk that if you take it off, you will not put it on, right? And we don't want the device to end up on your dog or something. We want you to keep it on. We have seen things like that before.
The device needs to be Bryant-proof, which means you can't lose it.
Yeah. Right. Skin friendly is also important.
So on that, on Kyle's note, make it FA proof as well. I used to have a Fitbit and I smashed it all the time. So keep in mind, hitting things, falling, it's got to be tough as well.
I see. Okay.
My question is, in your preliminary testing of this, your data is going to be basically collected without anybody knowing it. But what about knowing what is actually happening while you're collecting that data? Is there going to be a portion of it that you're going to have to enter manual data? Like I was walking, and this is what was going on during that.
Very, very good question. Like basically, if there is a diary that needs to be, to complement that type of data. The idea is that we won't need that. Part of the validation is to do some of these studies in simulated free living conditions. So you would, for example, come into some sort of a laboratory that actually looks like a suite in a hotel. You will have everything there. You will have kitchen. You will have a sofa, bed. You will have TV. You'll basically spend a couple of hours there while we are watching you, and then we will have the visual confirmation to know that what we collect is what we see, and we can put it together. After those algorithms are being developed and the machine learning is doing the job, we won't need that anymore. That's not part of the trial to bring you to a simulated laboratory. It's part of the development and validation of these devices and making sure that we get data that we can really trust because we're not there when it happens.
I agree with Kyle that having it be waterproof and sweat-proof and spill-proof would be great. I also am terrible with clasping. I can't put on watches. It's hard to put on necklaces.
Right.
So something that would be like if it fell off when you were pulling on clothes, or if it got caught on something and fell off, something that would be easily to put back on where you might not need help, that would be helpful.
Okay, that's a good point. What about recording your voice, being prompted to say sentences on a daily or weekly basis? How would you feel about recording something like that?
So earlier you had mentioned about like using it for clinical trials, and I feel like that would be really helpful for collecting data because when you go into the clinic, you might be dosing at the clinic at a different time than you are at home. So like measuring data differently, like at home it might be different than measuring it in the clinic, and I feel like that would be really helpful to have that.
So with respect to recording, voice recording or things like that, I don't think that is a major issue or problem. Both of my girls have been in trials with similar devices where they've had to do that, and it's just part of the protocol.
And it would work both if you had to read a sentence or hear a sentence and repeat it?
At the stage they're at, I think either is fine, but that might be different for different folks.
So everything you're saying sounds really good. I mean, about things these devices can do. Personally, I worry a little bit about the security, especially if they're going to the cloud or to like our personal devices, because I don't need someone overseas knowing exactly like how fast my heart's beating or anything like that. So is there some like security thing that you're thinking about? And since it's health information, how secure would it be, and would it be protected under HIPAA?
Yes, very good question. Thank you for bringing this up. I don't think that type of data would be different than any type of layer of security that we have normally on any other patient data that we need to protect. If the data in the clouds, there is a chance that God might know, but other than that, you know, it's supposed to be very, very protected. If there is a specific feature that you would like some of these devices to have that has nothing to do with Friedreich's ataxia, but something that would be cool for you to do, you know, for example, being able to get a message on the device, or being able to play music through the device. I'm just sort of brainstorming on that. Is there something that you can get as extra?
First off, I want to say everything that you mentioned in there, sign me up for. I will do every one of them. I've always thought, especially having late onset and I can still walk, when I go to the doctor and I have to walk down the hall and back, and they're like, wow, you're so good. I can't believe how good you are. In everyday life, I don't ever just have to walk down a hall and back and not pay attention to anything other than don't fall down. Walking around my apartment, I bump into walls. I don't do that at the doctor's office. And like she was saying, different times of day. It's completely different if I have a doctor's appointment at 8 a.m. versus 2:30. Speech-wise, you were asking reading something or just saying it. I have more trouble reading aloud. And I think it's when I went to speech therapy that processes, I can read it into my brain, but getting my mouth to form the words and whatnot is where I have the biggest problem. So actually reading something back and being able to measure that would be, at least personally, more beneficial.
All right, thank you. Thank you.
Thank you, and appreciate everyone really contributing to the discussion and asking good questions. We're going to kind of switch gears again and go back to talking about recent studies that have been done and trial results. And so Dr. Lynch is here with us today, and he's going to report on results from part 1 of the Moxy study for omaveloxolone. OMAV.
Okay, and does this pointer work or not? Pointer doesn't work. Okay, we'll do it however I choose. So thank you very much for letting me come today. I want to thank all the investigators in the study who are listed on this slide here in very small print that I cannot read because I'm having trouble seeing at my age. On the safety, efficacy, and the effects which we've seen in the recent trial of RT408, also known as omaveloxolone or something like that, or OMAV for short. As I'm sure everyone has become aware, if they did not know at the beginning of the day, Friedreich ataxia is a progressive neuromuscular and ataxic disorder which affects both children and adults. It's associated by a loss of the downregulation or loss of the protein called frataxin, which gives rise to a mitochondrial syndrome associated with fatigue, neuromuscular disturbances, and is truly a metabolic disorder as well. There are no approved treatments, as we all know at this point. with time, in recent clinical trials, as Bob and my previous speaker have mentioned, we have measured things through a quantified neurologic exam referred to as the Friedreich Ataxia Rating Scale, or FARS, originally designed by Sub Subramoni, who's now at the University of Florida. It includes sections which are essentially a quantification of a neurological exam. However, over time, we have adapted this so that it is really driven by patient-performed activities, where the neurologist-driven parts of this have been omitted, and it's now called the MFARS, or Modified FARS for short. Typically, people change around about 2 units per year. As you have on this initial slide, you see that over time, this score does worsen, and it correlates highly with the activities of daily living scale, which is a subjective patient-reported scale on how people are changing over time. In fact, it's the correlation coefficient is 0.84 in our large 821 subject natural history study called FACOMS. We're here today to talk about a little bit about OMAV, which is acts on as a transcriptional activator of the transcription factor NRF2, or NRF2 for short. This is part of the system your body uses to protect you against mitochondrial disease, and it's actually been shown that NRF2 and mitochondria are dysregulated in Friedreich ataxia. Since I don't have a pointer, I'll refer to the slides as well. I magically have a pointer if I whine loud enough. The red button. There, I got it. Okay. When you look at NRF2 activation status in Friedreich ataxia, the whiter the controls here, the blacker people with FA, you see that there actually is a particular deficiency of nuclear NRF2. That's where this transcription factor is activating on genes to enhance your response to mitochondrial issues. You can also look at some of the downstream gene products of NRF2, in particular one called NQ01, and they are markedly downregulated in people with Friedreich ataxia. In addition, people are mitochondrially abnormal in Friedreich ataxia. This is a graph of their peak oxygen consumption and exercise testing, which is a relative mitochondrial marker, and a correlation with their FARS exam. So in fact, the FARS exam does correspond with how people change over time. In this study, we are going to test whether OMAV can successfully induce NRF2 and thus induce the transcription of those genes which are downstream from NRF2, restore mitochondrial function, and perhaps look and see whether it can help patients as well. It has been shown in cell culture systems that in fact it can do this. These slides here are looking at NQ01 induction in muscle cells as well as ATP induction in muscle cells in response to the OMAV, the NRF2 activator. There's one thing which is crucial to realize, which you may not see in many studies, that as you look here at this, you see a very clear dose-response curve with a peak in the middle. This is characteristic of a tightly controlled process and really shows that what we are looking at is an appropriate biological response if one can reproduce this finding. You see here looking again in FA cells in a dish, induction of NQ01, as well as an effect on transmembrane potential, looking at mitochondrial transmembrane potential in a dish. So there is rationale that OMAV should be able to act on the NRF2 side and thus improve function in people with FA. This is the design of the study. It's in very small print. It's available on clinicaltrials.gov if you want all the details. But the key features of this is a 2-part study. Today you'll hear only about part 1. Part 2 is yet to occur. It looks at serial cohorts of 8 patients at a series of dose groups looking to dose optimize. At several of the doses they actually enrolled 2 cohorts as they got close to the important points on the dose response curve. 12 weeks of treatment, 3-to-1 drug to placebo, so you'll see in that group of 8 individuals, 6 are on drug, 2 are on placebo. Safety, of course, is the main component, and really looking for a signal in all of the other markers. It is not powered to demonstrate efficacy and prove that people should get better. It is proven powered to find a signal that can be amplified in further studies. We have multiple clinical assessments. The primary outcome measure is cardiopulmonary exercise testing. The secondary outcome measure is the modified FARS exam, measured at the beginning and the end of the study. There are a variety of eligibility criteria. Key components are people who were ages 16 to 40, and you had to be able to carry out the exercise bike riding at a reproducible speed. The questions which this study is designed to answer, which is always important to remind ourselves so that we know how to interpret the eventual results. Does OMAV induce NRF2 in FA patients? Is it in fact active? Does this induction translate to any improvement of metabolism and mitochondrial function, the basic cellular processes behind Friedreich ataxia? Is there an optimal dose, and can we replicate something resembling the in vitro dose to show that this is really acting on a target receptor? Does it improve any clinically meaningful measure of ataxia or exercise capacity, remembering that we are not powered to find a statistically significant effect, but looking to find a signal which can be amplified on the future? Are there people who respond more or less to the agent if we can identify any improvement? And also, is it safe and tolerable? This is the results from the randomization. Again, I recruited well. There's only a single discontinuation of drug and a person who had a rash related to it. One placebo patient also discontinued. At the doses of 160 and 300 milligrams, the highest doses, we double enrolled the cohort. That is, we took twice as many individuals there because that is the target area for where one might expect to start seeing interesting results. If you look at what the people were like who enrolled in the study, it's more or less what was expected. An average age of study entry of 25.6, so adults, sort of mid-range to adulthood. An average age of diagnosis of 15.3. So this is a slightly later onset cohort than the average FA patient who presents between ages 10 and 11. Realize that as we go in, that's what was selected for and that's what was expected. The individuals are largely ambulatory, they can, as defined by the fact they can perform a timed 25-foot walk. And they have a modified fire score about 41.1. Typically speaking, a person will become wheelchair bound or between 60 and 70 on that scale. So we're substantially on the, significantly on the less affected side on that particular measure. Does OMAV do anything to NRF2, where there are a couple endogenous blood markers that are controlled by NRF2 in everyone's body? One of them is ferritin levels. Another is GGT, which comes from the liver. And what you see is as you increase the dose of OMAV, you see a dose-dependent increase in ferritin levels, showing that this drug is likely hitting its target. In addition, as a parallel judgment, you can see an increase essentially within the normal range of GGT levels. This does not represent hepatic disease or anything like that, but it's in fact a biological effect of activation of the NRF2 transcription pathway. So this drug does, in a dose-dependent fashion, hit its target. Does it do anything to metabolism or mitochondrial markers in this study? AST levels, largely coming from muscle, but also from other tissues, are a marker of mitochondrial function through metabolic pathways. What you see is, again, a dose-dependent increase in AST levels, peaking out at 160 of the 160 milligram dose. At the same time, creatine kinase levels leaking out of muscle are another marker of metabolism. This is all within the normal range, so none of these numbers are actually pathologic in people for the most part. And you see, again, a dose-dependent decrease, peaking again at 160 milligrams, which is where it would have been predicted and where you saw, in general, the best activation of NRF2 in the ferritin and GGT levels. Does it affect people? These are the scores on the mFARS exam. I'll slow down just briefly so that people can understand this a little more. The way this slide is set up, this is the placebo group here. This is all people who went on drug, including the people who went on at very low doses, because this was a dose finding and you always start at a dose where you do not expect to see any effect. So you see placebo, OMAV, a largely dose-dependent improvement in mFARS exam. Down is improvement. This is about 4 points. This is a little bit, the mean change per year is less on that than on a full FARS exam, with, again, a fall off when one gets to 300 milligrams. So there is, again, that U-shaped curve, which would be typical of a biological target. When you look at the p-values across it and you only look at the 160 milligram dose, you can find a borderline difference with placebo and a significant difference from baseline in that group. And you see, if you look at these p-values, that those p-values are actually also dose-dependent. So as you get to the active dose, you do see an improving p-value and improving response. The data tighten up. You can also look at this another way and compare the response in the neurologic exam to the pharmacodynamic response. And when you do that, not on an individual basis necessarily, but on a group basis, the peak improvement in AST is a marker of mitochondrial metabolism, corresponds perfectly with the peak improvement in neurologic exam. This is displayed another way over here, and if you look here as essentially a responder analysis, the percent of patients who have a change of greater than 2.5 units, which is roughly 1 year's worth of change, it is concentrated 70% in those doses of 80 and 160 milligrams, exactly the target range one would expect for this drug. Is it time dependent? Yes, it is. The evaluation points were time 0, 4 weeks, and 12 weeks. And you see over time the response, the differentiation from placebo, as well as the improvement relative to baseline increases over time in each of the OMAV dose groups we've shown here. All of the groups, including both the low dose, which has little response, and the absolutely high dose, which has no response. But in those central groups, 80 curve like this and 160 like this. So again, it's time dependent and dose dependent. You would expect the effect to build with time. I will take a digression here and notice a little bit about the response here. This is a pretty typical placebo response. I will agree with Jen. I think most of what is called, quote, placebo response in FA studies is mainly practice. We can practice people into a neurologic exam. Try it sometime. We can practice people into an exercise test. Probably just about anything we do will have a significant practice effect unless it's so much a part of your daily life. I'm not sure a time 25-foot walk would, because you walk every day. You do things like that, so the practice effect will be smaller there. But this is a pretty typical placebo curve. Are there groups who did better than others? And when one looks at the data and tries to stratify across that, there is one group which can be identified. Musculoskeletal foot deformity is another name for pes cavus, those high arch feet which some people, in fact a majority of people with FA will have. And when you simply look at the data in this group, the individuals who had pes cavus did not show as much benefit as those individuals. The individuals who did have pes cavus did not show as much benefit as those who did not. And this is shown here that you in fact enhance the amount of difference in mFARS if you exclude post hoc the individuals who have pes cavus. What this means is not clear. You're selecting for a group, and whether it's a marker of something about the testing or something about the disease stage which is most effective is not really clear at this moment. But it is another way to help stratify the data as we move forward. This is another way to display the data where we've looked at on the top the mFARS response in all of the in a representative group of individuals, the 17. And then as we look down here at the key 80 and 160 milligram doses where you saw a response, people who have pes cavus are in gray, people who do not have pes cavus in blue. And you can see as you look at the comparison that the blues tend to go toward the left, which are the individuals who responded more, but that is not an absolute rule. This individual here, who I do not know who that is, showed a big response even though they did have pes cavus. So it is not a yes or no phenomenon. It is a relative effect which one can use either to enhance your sensitivity in future studies or to help understand the anatomic basis of what's going on. The primary outcome measure was exercise testing. When you look at exercise testing, there was no significant change in peak workload and the OMAV group compared to the placebo group or even compared to baseline. However, if you do exclude individuals who have pes cavus, you now can see a bigger effect. We're looking at the 80 milligram group and the 160 milligram group, as well as the whole group. You see an improvement over placebo in a change in peak workload, suggesting that there is a signal there, but we need to do some post-hoc calculations to enhance it and reveal that it is truly there. Other efficacy measures. We did time 25-foot walk, so I'll show you them very briefly. That is to say, if you look, you see some scattered p-values which are okay comparing to placebo. Essentially, you see the same effect again, where again, people with pes cavus had less benefit on the time 25-foot walk than individuals without pes cavus, and it doesn't reach statistical significance without that post-hoc exclusion. But you see the same type of data suggesting that there is a benefit here. pharmacokinetics, because this is a phase 2 study. Short story, since pharmacokinetics are really exciting, and I could talk about these all day, but you would be bored. Yes, the more drug you give, the more you see in terms of levels, and you do see some diversity, but it is reasonably reproducible and controllable. The numbers are not all over the place, which is good for moving forward should that occur. And perhaps the most important thing, adverse events. This drug was safe. There are only 2 serious adverse events, both of which occurred in placebo patients. One being benzodiazepine withdrawal, and the other person had 3rd-degree burns for an event unrelated to Friedreich ataxia. I actually know that subject. And the adverse events that were seen were really basically mild. Headache, ligament sprain, very small numbers of individuals, so this is a safe drug within this modest size study of about 50 people on active drug. So overall, I think this is a relatively positive result. In fact, you might say it's probably about as positive as a phase 2 study could be expected to be. If you go back to the original questions we asked, did we induce NRF2 in Friedreich ataxia? Yes, because GGT levels went up and because ferritin levels went up. Now let's take one other digression. What I don't think people realize is that ferritin actually is a biomarker of Friedreich ataxia. With the lower your ferritin level is, the worse your Friedreich ataxia level is. The worse your Friedreich ataxia is. So in fact, you could claim that this reversal of ferritin is reversing a disease-selective biomarker. So you are hitting your target, and you are reversing something which is known to correlate with disease severity in FA. In addition, we see some response in CK levels and AST levels to show that it works on metabolism and mitochondrial function. There's some modest increase in exercise capacity at a single dose, and improvement in mFARS exam at the key doses here, an acceptable adverse event profile across the board with only a single discontinuation related to rash. So this is overall what I would call a successful phase 2 study in demonstrating how to move on with more pivotal studies moving forward. Now, this study was written originally as a 2-part study, with the first being designed for 3 months and the second being designed to go 6 months or longer in order to refine the precision of the approach. This is planned at this point and for upcoming months. The only differences from the part 1 are essentially the outcome measures are flipped, with the mFARS exam being the primary outcome measure and the exercise testing being a secondary outcome measure. And the other thing which is changed is the duration of therapy goes from 3 months to 6 months, getting closer to what would be a goal being a registration type event or something like that. So I think this is actually a very successful phase 2 study, and that gives me the chance to thank everyone who participated in the study, including a large number of individuals in this room, without whom this study would not have been possible. And I'll take any questions you have. Mr. Conans.
Do you feel the need to look between 160 and 300 to see if it continues to peak before it falls off? What's your thoughts there?
So that's a very good question. I don't think it's really very easily done. It's very hard to control. Usually, twofold differences in drug are pretty precise. I wouldn't think that you'd be able to get much mileage there. You've already hit the peak. Could you do better at 200? You might, but across a population, I don't know that you could ever control levels well enough, because there is some variability between, let's take presumably normal people, me and Jen, and we would simply metabolize it different. Paul is rolling his eyes about us being normal people. The individual metabolism would be sufficiently different in a way which would be very hard to determine, that I don't think you'd ever be able to determine that precisely enough. So I do, right now, the targeted dose in the next group will be the 160 milligram dose. Gio.
Thank you, Dave. Can you explain why CK and ST are markers of mitochondrial function?
So the question is why the AST and CK are markers of mitochondrial function. For AST, it's because of its role in shuttling components through the malate shuttle to provide more energy for the mitochondria itself, so it's a secondary metabolic marker. For CK, it, I believe, is a presumed leakage that related to subtle mitochondrial dysfunction. CK does have a couple confounders. The biggest confounder in FA population, anyone know what it is? It's falls. The more you fall, you hit your muscle, and you leak CK. And it's going down, so if that is what the underlying marker actually is, that's a good thing as well. Mr. Bryant.
So these are pretty cool results. So now what? What's next?
There.
Oh, thank you.
Yes, there is a plan to move on. Still, the final details are still being worked out at this moment.
Could you explain to us the significance of foot deformity in this study? Is that a marker of underlying disease? Is that an exercise outcome measure?
So the question is, what is the significance? And I'll go to its usual name, pes cavus, because I'm so used to it. That's a good question. Why do people with pes cavus have less response? I'll give you three possibilities. The most important one is it a marker of something else which it simply is correlated with? I think that's in a small study, and yes, 50 studies on drug is a relatively small study. You never know whether you actually picked up something of a corresponding variable, which is not that. The second possibility is the outcome measure is an exercise test. And there you saw that they did more worse, if that is a phrase in the English language, than in the MFARS exam. Is it possible because you do have to fit pedals? You know, can they just not give enough effort? I will note one thing which is unusual about this study is the way we designed it, is that everyone is measured an hour after, for their MFARS, an hour after their exercise test. If the amount of fatigue you induce in the exercise test gives us those really tight numbers that we see, then how much you exercise in the test actually could influence your response down the road and the amount of placebo and practice effect you have. Could it be something like that as well? I'll give those two possibilities. The other possibility is, to get a little bit more Friedreich ataxia right, is you think about the neuroanatomic basis of Friedreich ataxia. We have learned that it is more than just dorsal root ganglia, that involves central nervous system pathways, it involves metabolic myopathy. The pes cavus tends to occur early, and it is probably a marker of more developmental components that logically may not be as responsive looking down the road. So it could be a marker of the disease phenotype, and that's what we've picked up upon. Jen said early in the day that we imagine using multiple drugs to treat this, and that is, while the effect here is extremely encouraging, maybe hits one part of the disease, which we will certainly take, and I think that's a message you've heard throughout the day, is to pick one symptom, or we may have different drugs, and this picks up one, and the pes cavus may be alerting us to one particular one. And I will note, I favor the anatomical hypothesis, but I cannot prove it.
Hi, Dr. Lynch. For part 2 of the study, will it be the same age range, or will we be looking at younger patients as well?
So in the part 2, the age range will be the same. It's still a part of the original inclusion criteria. 16 is the lower limit on age. I will note that we were only allowed 18 at CHOP, which we will be arguing with our IRB. Yes, Felicia in back.
Dave,
yeah.
Quick question. When you say that there are significant improvements in the FAR scale, is it across the entire scale, or there are things that just don't improve and only part of it is improving?
A good question. Are there subcomponents here? So remembering that the MFARS is a little bit stripped down. That is, it's taken out the peripheral nerve component, and it's taken out the speech and bulbar component, so it is really an upper limb exam, a lower exam, and then the standing and walking component. Is it possible that there's particular subcomponents respond? Yeah, we have to go back and look at that, but again, to see if it's present on any, if there is a subgroup, a subcomponent that the response is in, one of the problems is, again, 50 subjects is a little small to start doing it. So there'll be some data on that, whether it will be conclusive either way, I can't say.
Dave, another question here. Sorry. So I don't see any biochemical or target engagement measures in the phase 2, like
okay.
frataxin levels in buccal swab. Supposed to go up, right?
So within this is the general synopsis. There are biochemical measures that are built in. The same ones as before. A point of discussion will be to whether to measure frataxin levels. In some in vitro studies, frataxin expression itself is downstream of NRF2. The hardest thing is about measuring frataxin levels in clinical trials is getting the correct target tissue. There's some pieces of data from this study that are still pending, including mitochondrial metabolism function from Ian Blair's lab, as well as the results of frataxin testing from buccal swabs. I would say, I'll say it right now, across several studies, across the natural history study, I do not believe that blood frataxin levels are of much, whole blood levels are of much meaning, because I think the regulation is substantially different. The other place to go is muscle biopsy, and there were a limited number of muscle biopsies within this study for which we do not have results yet.
So what about oxidative stress markers in blood? Those you can measure, right?
The question about oxidative stress markers in blood, I would, while that remains open for discussion, we've never gotten an oxidative stress marker really to work very well in blood in the baseline population, so I don't know that it would be something in which we can conclude things. You will recall at least 2 clinical trials before that were confounded when it was elected to measure urinary dihydroxyguanosines, and they were found not to be abnormal from the control population, thus making their interpretation problematic. I actually think what has been derived in this test is markers of activity. And I'm going to stress that as I look at the data at the end, and this is a post hoc conclusion on my part that was not on the slide because I didn't make it till earlier today, is that I think the ferritin levels, because they correlate so well with genetic disease severity in FA, I think it is hitting the biological target dead on. I'll say that on the record. Yes, Ron.
Dave, going back to the Pescavus question one last time, you say you favor the anatomical explanation.
That's me, but other people in the room and elsewhere may not.
I personally agree with you. I think it might be the...
Good, I love you, Ron.
Yeah, I love you too. It might be just the inability to work the pedal as well as...
It may, and also may be what most things are.
Yeah.
Multifactorial.
Exactly. But just to the point, I know there are people in the room whose patients or the patients themselves have Pescavus.
We're looking at that right
They
now.
would want to know, I think, because you would confirm, there's no reason to believe with those results that a patient with Pescavus would not be a beneficiary of this treatment if it proves to be effective. I mean, you would think
this is a bit complicated slide. If you're not used to reading it, it may be. Look at the bottom here and look at the mFARS response. Yes, the people with the highest response are in blue, but this person's in gray. That's a response of about 8 points, and that would actually be higher than any placebo. In fact, that person in gray and that person in gray would be, and that person in gray would be higher than any placebo person except that. So the data would say it works across both groups, but it's just a shift in the curve and the relative sensitivity of us to find it at this point. We don't know the full interpretation. I know, I just love your feet, but I don't know where that came from. If we ask any more questions, who knows what Dave will say. One back from Gretchen, it looks like.
I just had a question about one of the exclusion criteria for the heart significant cardiac disease. Is that because of something of the drug or trying to make it homogenize the samples?
So the question is the exclusion of cardiomyopathy. Let's step back a minute and think about the concept of exclusion criteria to begin with. I think there are 2 reasons. One is safety, particularly as you go into an unknown. And while I know of no reason to believe that there's an unsafe event here, it is an unknown, and your job is to do no harm first. So until we are sure, I think you have to be very cautious. The other thing is when we and this gets to what our previous speaker was talking about. When we do trials, we set it up in an idealized situation, which has its benefits and its issues. The more diversity we have, the harder to know to make it interpretable, particularly in a first study. So homogeneous populations use a lot smaller sample sizes than heterogeneous populations. So I do not anticipate that exclusion criteria changing.
Okay, that's what I thought. I thought maybe also it was the exercise, and you were just a little worried about it.
Yeah, exercise, of course, in FA, what is an exercise measuring? I think it measures lots of different things, which is both good and bad.
So if you go to just if I could add on, if you go to a phase 3, say the second part comes out really good, and you go to a phase 3, would you then maybe drop the cardiac exclusion?
I think we have to look at the diversity of what goes forward, because I think one of the things we talk more and more about is in textbooks you see phase 1, you see preclinical phase 1, phase 2, and phase 3. I think there are a variety of pathways to move forward, particularly in rare disease, so that I don't know what the method forward will be at this point.
Okay, thanks.
And Kyle has one more. Things are being worked on right now, and we'll see how things move forward. It's officially, babe. Thanks.
So I'd like to thank all of our speakers this afternoon. We covered a lot of topics. I appreciated Amir getting us out of our normal box and thinking about how to leverage new technology for future clinical trials. I think that was really exciting. And I would like to thank Jupiter and Retrotope and Reata for sharing their work in advancing new treatments. Continues to be really exciting, having a diverse treatment pipeline. And with the results from the RT001 study, we see, a novel outcome measure, the exercise test, showing improvement, which was really encouraging. I think Dr. Lynch made an important point. These were early phase studies that were not powered to show efficacy. They were powered to show safety and dose. And so the fact that we're seeing efficacy signals in placebo-controlled trials at these early stages are incredibly encouraging. And then, the RTA408 or omavaloxolone study also showing us positive results in the MFARS is also really exciting, and I think it's a nice way to end our day. And so I thank you all for being here and for your attention. I know it was a long day, but thank you. Oh.